Dear respected participants, welcome to our second International Congress. I am Associated Professor Ulaş Acaröz from Afyonkarahisar University Faculty of Veterinary Medicine. Today is very special day for me because we are organizing second time a global conference. In our first conference, there was attendance from 21 different countries. And today we have succeeded to get this support from 50 different countries. I appreciate everybody here for their support to our Congress. And also, we have organized this Congress with Professor Dr. Kui Su. We have made our doctorate in Munich together in Germany. And the first invited speech is going to be done by our supervisor, Professor Dr. Mertel Bauer. I appreciate him for everything that he done for our academic life. And we can start with our first sorry for the inconvenience. My microphone is closed.
Organize edilen 2. Uluslararası Biyoloji ve Sağlık Kongresi'ne hoş geldiniz. Düzenleme komitemize bizleri bu çatı altında topladığı ve bilimsel bir tartışma zemini oluşturduğu için teşekkür ederim. Bildiğiniz üzere tek sağlık konsepti beşeri tababet yanı sıra veteriner hekimleri de kapsamaktadır. Malumunuz özellikle zoonoz hastalıklar dünyada önemli bir halk sağlığı sorunu olarak karşımızdadır. Dolayısıyla tek sağlık konsepti de işte bu çerçevede önemlidir. Tüm dünyayı etkisi altına alan COVID-19 pandemisi tek sağlık yaklaşımının önemini ve gerekliliğini bir kez daha vurgulamıştır. Ülkemizde pandemi sürecinde hastalığın yayılımını önlemek, tabii ki başta aşı geliştirmek, tedavi için çeşitli ilaçlar, antiserumlar üretmek üzere farklı meslek grupları tek sağlık çatısı altında özverili bir şekilde günümüzde çalışmaktadır. İşte tüm bu doneler tek sağlığın ve tek tıbbın önemine vurgu yapan unsurlardır. Tek sağlık konseptinde yer alan çalışmaların ve güncel konuların saygıdeğer araştırmacılar tarafından ele alınacak olduğu kongremizin başarılı ve verimli geçmesini diliyorum. Selam ve saygılarımla.
We appreciate to the Dean of Afyonkarahisar University Faculty of Veterinary Medicine. I would like to appreciate to our President, Professor Dr. Mehmet Karakaş, for his support for the realization of this Congress. And second speech is going to be held by Associated Professor Dr. İlhan Aydın, who is the Deputy Director of General Directorate of Agricultural Research and Policies, Turkey. Dear Dr. Aydın, it is your turn, please.
Thank you, dear Ulaş. First of all, I would like to thank to all colleagues they joined this very important event. So I am so happy to be part of this really important international organization. Let me say something for introduce to my institution and myself. The first institution was established 1894 in Istanbul. Now we say research for Pendic Research Institute for Veterinary Medicine and Veterinary Health. And then the many years we established the other institute, and now we have 48 research institute and 24 authorized research institute. The Veterinary Research Institute under our research, the organization, they had the capacity, as you know, to fight disease under the umbrella of One Health. We are assuming like this. And also, let me say, those research institute has the washing capacity. You know, these certain days, because of the COVID, we had some really difficult days, but I am really appreciate that we are honored to have that research institute. They have a capacity to produce to washing for not for the animal diseases, but also for the same capacity for human health. So on behalf of this research institute, I would like to say they have a really big capacity, and also we had 3 private companies which supported by our organization to produce the medicine or washing to fight the disease. Because of our really strong support through the project, those research institutes not only for belong to government, but also for those belong to private company has capacity to fight all disease occur in around Turkey. And also we are export to some washing to neighbor country to fight some disease. So let me say this research capacity in Turkey for those disease is very big, and we would like to support this capacity in near future as well. And also, this Congress, this organization is very important to share our knowledge to you and the colleagues. So I am so happy to be a part of this event, and I would like to say to fruitful the organization, all of you, and we are happy as a TAGEM, the part of this organization. I am so happy to thank to you and your colleague to have fruitful meeting. Thank you.
Dear Associated Professor Dr. İlhan Aydın, we appreciate for your presence and contribution to the Congress. The next speech is going to be held by Professor Dr. Kuyusu. We are organizing Congress together, and as I said at the beginning, it is a special day for me. Kuyi, it is your turn.
Okay. Hi, everybody. Good morning to each other. And I'm Kui Zhu from the National Center for Veterinary Drug Safety Evaluation, College of Veterinary Medicine, China Agriculture at Beijing. It's my great honor to be here to meet some old friends and to see more new friends. That's fantastic. So compared to COVID-19, I think antimicrobial resistance has become also a global crisis and on one health and the world economic as well. That's why I want to continue to call for the action in this second international conference. I believe in One Health approach to this complex problem. That means closer cooperation between human and the veterinary side and between the different research instruments that we have. One important solution set is the development of new antibiotics, alternative treatment and prevention options, such as vaccines, probiotics, to target therapies. Scientists from different research areas, from academia and industry, and from different countries work together to develop such new ways to combat bacterial infections. So it's important that we keep working together, share knowledge, define a shared strategic research agenda that reflects public health or One Health need and the urgent threats by a close connection to One Health need and shared strategy and choice, so we can compete the rapid emergence and the dissemination of AMR. So I wish everything goes well with all of you, and hopefully we will meet physically in the near future. Thank you and enjoy the conference.
Thank you very much, Kuya. It is nice to do something together. Sorry for the inconvenience. My microphone is closed by automatically. Kuya, thank you very much again for your valuable contribution to the Congress. And it is very nice to do something together. And
yeah, thanks.
And the next speech is going to be held by Professor Dr. Alparslan Ceylan, who is the rector of Kyrgyz Turkish Agriculture Manas University. I'm opening his video.
Takip eden, katkı sağlayan çok kıymetli Afyonkarahisar Üniversitesi tarafından düzenlenen Biyoloji ve Sağlık Bilimleri Kongresi'ne hoş geldiniz. Afyon Üniversitesi Rektörümüzün katkılarıyla düzenlenen bu kongre 50'yi aşkın ülke tarafından katılımla yapılmaktadır. 400'ü aşkın bilim insanının katkılarıyla düzenlenecek bu kongrenin başarılı geçeceğine hiç şüphemiz bulunmamaktadır. Bu kongrenin düzenlemesinde özellikle doçent doktor Ulaş Acarus'un önemli bir çalışmasının olduğunu biliyoruz. Afyon Üniversitesi'nin değerli hocalarının önemli katkıları olduğunu biliyoruz. Dolayısıyla bu sempozyum, bu kongre bilim dünyasında önemli bir açılım sağlayacağına hiç kuşku duymamaktayız. Sağlığın bu kadar önemli olduğu bir dönemde yapılan bu sempozyumun sonuçları bilim dünyasına önemli katkılar sağlayacaktır. Bu kongreye katkı sağlayan bütün bilim adamlarına sonsuz teşekkürlerimi sunuyorum. Manas Üniversitesi adına bu kongreyi takip eden, katkı sağlayan çok kıymetli hocamız, yardımcı doçent doktor Fatih İstanbullugil'e ve arkadaşlarına da teşekkürü bir borç biliyorum. Tanrı Dağları'ndan bütün katılımcılara gönül dolusu selamlar, saygılar.
We appreciate Professor Dr. Alparslan Ceylan for his contributions. And the next speech is going to be held by Professor Dr. Erinda Lika, who is the Vice President of Agriculture, University of Tirana, Albania. Dear Professor Dr. Erinda Lika, we are ready to listen you.
Hello, can you hear me?
Yes.
Hello, everybody. Hello, dear colleagues, deans, vice rectors, and directors, and thank you for the invitation, Professor Ulash. It's a pleasure for me today to be part of this opening speech of this very important conference, because talking about biology and talking about health science in the concept of One Health is a very important issue in our day's focus. Let me introduce my institution, my university. Agriculture University is the unique university in Albania. It is in Tirana district, and it has 5 very important faculties, but I have to mention only 2 of them that are closely related with health or with public health. So is the veterinary medicine faculty, which is also unique in our country, and also biotechnology food faculty. We have also capacities, human capacities and lab capacities to have creating the chance to have a good collaboration with all of the research institutions that cover the field of public health and cover the field of medicine science and also of food biotechnology science. So let me wish a lot of success for your Congress with very important issues that you are offering regarding the research and new scientists, and congratulations. Thank you very much.
Thank you very much, Professor Dr. Erinda Lika, for your contributions and presence. The next speech is going to be held by Professor Dr. Mikhail Ivanovich. He is the president of Vitebsk State Academy of Veterinary Medicine in Belarus. On behalf of him, Dr. Dmitry Mozorov is going to make the presentation, who is the Belarus country representative of FAO, and he is also very important work in the OEA.
Morning, dear colleagues. Can you hear me?
Yes, of course, we hear you.
Dear colleagues, I am representing here the Vitebsk State Academy of Veterinary Medicine located in Belarus. Unfortunately, director of our academy, Professor Khavrichenko Nikolai, was not able to participate at this event. He forwarded his warmest greetings to all participants of the Congress. He wishes that this event was successful, useful, and will be able to lead to development of wider network of specialists from different countries and organizations involved in veterinary medicine, public health, food and feed safety, and other topics which are parts of the concept One Health. Here in Belarus, we pay high attention to One Health concept because we see the challenges: globalization of trade, climate change, new diseases outbreaks, changes in human behavior, all of which provide multiple opportunities for pathogens to colonize new territories and evolve into new forms. The risk is not only for humans. Animal health is also greatly impacted by diseases passed from humans. It means that veterinary services, wildlife authorities, and researchers from universities must cooperate effectively. One individual country is not able to do anything alone. That requires a regional and global approach and partnership. Regarding Belarus, we are an export-oriented country. Our food export is about $7 billion. Regarding milk and milk products export, we have 4th position in the world. We have very high density population of animals in our farms. It's traditional for us to have few thousand cattle in one farm. We fully understand what can happen in case we will not follow this One Health concept. We had in the past outbreaks of transboundary animal diseases, and we got great experience how massive stamping out of animals can pollute the environment and negatively affect people's livelihood. With this goal, We tightly cooperate with international organizations, such as the World Organization for Animal Health, OIE, Food and Agriculture Organization of the United Nations, FAO, and WHO. This organization developed international standards regarding One Health. At national level, we try to implement these standards. As an example, we participate at regional and local technical cooperation projects on transboundary animal diseases with high economic impact, antimicrobial resistance in agricultural sector, and food safety with focus on foodborne diseases. Within this project, we reform our veterinary and food safety legislation, conduct trainings for staff of veterinary service and faculties, increase our common capacity, as well as establish new cooperation with the colleagues from abroad, exchanging our experience. At our educational establishment, Vitebsk Veterinary Academy, the staff is also involved in this project. We are planning to introduce new module on One Health for our students at all faculties. Recently, under OIE and WHO umbrella, human medicine and veterinary medicine faculties in former post-Soviet countries started to introduce One Health modules. And Belarus doesn't want to delay this process as well. Let me one more time, on behalf of our rector, from myself, thank organizers of this Congress, our Turkish partners and friends, especially Dr. Ulash, who visited us recently in Belarus and invited to participate in this important event. We very hope that next time the Congress will be able to conduct the face-to-face format. Thank you for your attention and all the best.
Thank you very much, Dr. Dmitry Mozorov, for your valuable contributions. It was also very nice. We have listened your experience about the PAO also. Thank you very much again for your contributions. And next speech is going to be held by Professor Dr. Mehmet Emin Erkan, who is the president of Şırnak University. Hocam söz sizde.
Merhaba arkadaşlar. Hepiniz 2. Uluslararası Biyolojik ve Sağlık Bilimleri Kongresi'ne hoş geldiniz diyorum. Kongrede emeği geçen bütün arkadaşlara saygılarımı sunuyorum. Şimdi tek sağlık konsepti ve multidisipliner çalışmalar önümüzdeki yıllarda önemini kat kat arttırarak devam edecektir. Özellikle koruyucu hekimlikte insan sağlığı, hayvan sağlığı ve bütün birimlerde bu aşıyla ilgili çalışmalar önemini daha da ortaya koymaktadır. Burada da veteriner hekim arkadaşlarımızın, işte Türkiye'de Türkovak aşısının yapımında Yakut Darendeli hocalar, Osman Erganiş hocalar ve dünyada bütün Çin'de de Amerika'da da multidisipliner çalışmalarla veteriner hekimlerin olduğu, tıpçıların olduğu, biyologların olduğu, kimyagerlerin olduğu ünitelerde aşılar üretiliyor. Bu da bize tek sağlık konseptinin ne kadar önemli olduğunu, çözüm üretmek için, dünyayı sağlığa kavuşturmak için bunun ne kadar önemli olduğunu göstermektedir. Kongremizin hayırlara vesile olmasını temenni ediyor. Bütün katılımcılara saygılarımı sunuyorum.
Thank you very much, Professor Dr. Mehmet Emin Erkan, for your valuable speech in which the importance of the One Health is especially stated. Thank you very much again for your contributions. The next speech is going to be held Professor Dr. Talat Nasir Pasha, who is the president of University of Education, Lahore, Pakistan. We respected professor, we are listening to you.
Bismillahirrahmanirrahim, and greetings from Pakistan to all the respected rectors, deans, organizers, and it's indeed a privilege for me to speak on this August gathering and very important conference on biological and health sciences. As a matter of fact, people, animal, and environment are very important in this One Health subject, and due to very extensive and mass communication among countries, We have recently noticed the COVID, how it spreads across the world. And we, as a matter of fact, have to work together and for emerging and re-emerging diseases and viruses, and for the food safety and the foodborne diseases and all other issues which are involving trade and which are involving in the health of humans and animals. And in this country, at our University of Veterinary and Animal Sciences, it worked a lot during this COVID, and they at their BSL level 3 lab, they volunteered themselves for conducting of the COVID test, and over 700,000 tests were conducted so far, and developed COVID vaccines also. So how this is veterinarians and health professionals have to work together to face the challenges of disease and face the challenges of foodborne issues and other. And I have gone through the program of this conference, which is very nicely arranged, and it will provide an excellent opportunity for learning from each other and sharing experiences. And I must congratulate all the team, Professor Ulash and Rector Afyan, and all the honorable leaders for arranging this wonderful conference. And we look forward that next time we will meet face to face and have a more opportunity to learn from each other. Thank you very much. And I'm obliged for this opportunity.
Dear Professor Dr. Talat Pasha, thank you very much again for your valuable contributions. We have completed now our opening speeches. I individually thank for every speakers for their valuable contributions for the opening ceremony. And we will start now with the first presentation. He is very special person for me and for QUI, because he is our supervisor. In Germany, supervisor is called like father, and he is our doctoral father. And Professor Dr. Erwin Mertelbauer is here. And thank you very much, Professor Mertelbauer, that you are here. And I would like to also thank you very much. He is most probably not here, but Dr. Dietrich, he has contributed also to us a lot. And we are happy to listen to you there, Professor Dr. Erwin Mertelbauer.
Yeah, thank you very much, Ulash. Good morning, ladies and gentlemen, dear colleagues. It's a great pleasure for me to participate in this conference, which has organized by my former students. Thank you very much, Ulash. Thank you very much, QUI, for inviting me, and my congratulations for the organization of this very, very, very interesting Congress. So I think I have to share my screen with you.
Yes, I have opened to share screen option for you. Firstly, you have to open your presentation. Afterwards, at the bottom side, there is share screen, and then you will select the presentation from the section.
Just a minute, I think my so let's try. Can you see my screen?
Yes.
Okay. Seems to work, okay?
Everything is fine.
Yeah. Well, good morning again. And today I am going to talk about foodborne bacterial toxins. As you know, foodborne diseases are a major cause of human illness. And an expert group of the World Health Organization calculated that it causes millions of illness and several hundreds of thousands of deaths per year and enormous economic losses. And besides foodborne infections caused by viruses, pathogenic bacteria and their toxins are of major concern. In this talk, I will focus on foodborne intoxications. That means illness caused by bacterial toxins, which have been formed in the food and act immediately after arrival in the intestine. This also means that food intoxications can have very short incubation times of 1 hour or even less. And the most common causative agents for food intoxications is what you can see on the right side of the first slide. Toxins produced by Staphylococcus aureus and Bacillus cereus. In the European Union, EFSA, the Food Safety Organization, calculated or reported several outbreaks and a lot of individual cases for the year 2019. And you can see in this slide that, that the number of cases caused by bacterial toxins, which is above 10,000 per year, is exactly in the range for infections which have been caused by Salmonella, for instance. And the most of these cases, or more than 50% of these cases, have been caused by Bacillus cereus and Aureus, or by toxins produced by this species. Let me give a short introduction to Bacillus cereus. It is a gram-positive rod which is motile and grows aerobic but also anaerobic. It is a common soil saprophyte and is easily spread to many types of food, particularly to food of plant origin, but it's also frequently isolated from meat, eggs, and dairy products. In the so-called Bacillus cereus group or cereus group, currently 9 species are listed, including Bacillus anthracis and several others. And all of them can produce, or most of them can produce specific toxins. But the hematic toxin I'm going to talk about today is only produced by a certain subgroup of Bacillus cereus. Well, Staph aureus, it's also a gram-positive bacillus. It's a coccus, it's non-motile, and can also grow aerobic and anaerobic. And the toxin-producing species are all so-called coagulase-positive species. And among them, the main important one is the subspecies Aureus, but also some strains of Staphylococcus intermedius, Huicus, or Delphini can produce these so-called enterotoxins. You can see on this slide that beside these illness caused by the toxins, both species can also cause a number of non-gastrointestinal diseases like septicemia, endocarditis in humans, or Staph aureus, well known for causing the so-called toxic shock syndrome, wound infection, osteomyelitis, endocarditis, impetigo, and so on. And also they can cause several diseases in animals. For example, Bacillus cereus can cause mastitis in cattle. Also Staph aureus, which is one of the major causes of mastitis in cattle, and several others, and more or less unspecific infections. But today, what I'm going to talk about today is the gastrointestinal diseases caused by these species. And if you have a look on this slide, you can see that the symptoms are very, very similar. Both Staph aureus and Bacillus cereus cause an illness which is characterized by emesis, also emetic symptoms, and partly by diarrhea. The time to onset of this disease is also very similar for these species. It's in the range between 1 and 6 hours, and there are cases reported where it even was less than 1 hour. The only difference is, as far as we know today, that there is a difference in the toxic dose. As you can see here, for Bacillus cereus intoxications with the emetic toxin, 3 to 30 micrograms per gram food have been reported from individual cases as toxic dose, where for Staph aureus enterotoxins, this dose is much lower. It's in the range between 0.1 and 0.4 micrograms per person, which is indeed much lower. And this has been estimated, for example, for Staphylococcal Enterotoxin A in the 60s of the last century. And these are very exact figures we know. For Bacillus cereus, we have only data from food intoxications where the amount of toxin has been analyzed in the respective food. Also, as I mentioned before, very important, the toxin of both species, the emetic toxin and the Staphylococcal enterotoxins, are produced in the food, already in the food. And as soon as a consumer gets this food, the symptoms, the toxins reach the intestine, they can provoke their action, and this explains the very short incubation time. Let's start with the emetic toxin of Bacillus cereus. It's so-called, it's called cereolide, and the original one you can see on the left side of the slide. It was isolated in 1994 by a Japanese group, and later on several isoforms of this cereolide have been described. So far, all named or got some letters in addition. So those are the isocereolides A, B, G at the moment. And and they show this typical ring structure. They have a very low molecular weight of about 1,200 Daltons, which renders them extremely stable to all procedures applied in food producing. And also this structure is quite hydrophobic. It's, as I said, very resistant to heat. and cannot be destroyed by enzymes in the digestive system. It has been shown in an animal model that this emetic toxin stimulates the vagus nerve and activates the 5-HT3 receptor, and thus triggers the vomiting center in the medulla oblongata. The symptoms of this emetic disease usually decline after 24 hours, but also some more severe intoxications with lethal outcome have been reported. Due to its high ionophoric activity, cereulide can also lead to liver damage and serious multi-organ failures. In cell lines, as you can see here on the slide, the mitochondrials are impaired. They begin to swell, and this mitochondrial dysfunction is greatly reflected in the reduction of maximal cell respiration. And the effect you can see on the two pictures on the right side. On above, you can see a cell layer which is not affected, and below a cell layer which has been affected by the emetic toxin. And you can see a dramatic loss in the number of cells in their shape. There have been, over the last 20 years, I think there have been several attempts to develop a detection method for this toxin. And in the meantime, in principle, 3 methods are applied. First, PCR or real-time PCRs. The first method developed was cell culture methods, using the specific action of this toxin on the cells as a readout. And also in the meantime, LCMS methods, as a mass spectromic methods, have been applied, and there is even since a few years there is a European standard method for the detection of this toxin. Unfortunately, commercial assays to detect this toxin are currently not available. And here a typical intoxication caused by the emetic toxin. It was recorded in a restaurant in Berlin. And that is very often the case. This Bacillus cereus food poisoning has often been connected to consumption of rice in restaurants. And it involves a common practice of bulk cooking of rice, and portions of this boiled rice are stored. And that's the greatest problem. If they are stored at room temperature for enough time, then Bacillus cereus can multiply. And as I told you before, the toxin itself can not be destroyed by additional heating steps. And if this rice is used for preparation of dishes the next day, and are offered to the consumer, then it will cause intoxication with the typical clinical symptoms. And in this case, the rice sample, when analyzed, contained Bacillus cereus up to 10 to 7 colony-forming units per gram of rice, which is quite a high amount, and certainly enough to produce enough toxin to cause food intoxication. Well, Let's switch to the enterotoxins produced by Staphylococcus aureus. There are in the meantime quite a huge number of these enterotoxins described, which are divided into the so-called classical enterotoxin types, which are enterotoxins A to E. And these enterotoxins are or have in common that all are proteins with molecular weight around 27 kilodaltons and astonishingly heat stable also. All these are proteins. They consist of two domains, have a disulfide bridge, and are so-called super antigens. And the cause emesis or the emetic action only in primates. Though that has research on this toxin made quite complicated because there is no real small experimental animal to make research on the mode of action and so on. The emetic action only occurs in primates. And it's also similar to the emetic toxin of Bacillus cereus. It's assumed that also the staphylococcal enterotoxins activate or serotonin and the 5-HT3 receptor. The current model of the mode of action is like this here, and it's assumed that the enterotoxins were taken up by epithelial or goblet cells in the intestine. After passing these cells, they should or shall activate mast cells, which release serotonin. And this serotonin stimulates again the nervus vagus, and this results in the emetic response of these toxins. It has been discussed for a long time if the so-called super antigenic action of the staphylococcal enterotoxins can have a role in this mode of action, but there is no clear indication that this is really the case. Well, I mentioned that in the meantime, we have a number of these staphylococcal enterotoxins described. And the first one, which was staphylococcal enterotoxin B, was isolated in 1959 and shortly followed by the other ones, A and C. And later on in the 70s, staphylococcal enterotoxins E and G have been described. And so far, as you can see here, we have, I think it's, if I calculated right, it's 25 of these enterotoxins. And the ones you can see here in red are enterotoxin where the emetic action has been proved by animal experiments. There are several others which have been described as staphylococcal enterotoxins, but based on molecular biological methods and similarity calculations. And all of these for which the emetic action has not been proved in animal experiments are called Staphylococcus aureus enterotoxin-like toxins. And this is the L you see in the name, and this is also quite a number of those toxins. Well, because these toxins are proteins or exoproteins, the method of choice for detection is antibody-based methods. So in particular, enzyme immunoassays. And there are several of these immunoassays on the market. This is just a selection of the most well-known companies you can see here. And the problem all of these commercial methods join is that they only detect the classical enterotoxins, A to E. Some of them can detect the individual toxins. Some of the assays just give a sum of toxins as a result, but none of these commercial assays can detect the one of the members of the new so-called new Staphylococcal Enterotoxins, which makes diagnostics measures a bit complicated at the moment. Yeah, finally, I wanted to present one of the most severe outbreaks caused by Staphylococcal Enterotoxins. It happened in a dairy plant in Japan. And somehow the milk the dairy plant acquired was contaminated with Staphylococcus aureus. And during storage of this milk, there was an electricity failure in the company, which led to the storage of this milk at a temperature between 20 and 30 degrees Celsius for 9 or more than 9 hours. The company was aware of the contamination of the milk with Staph aureus, but thought if they would heat the milk after this storage period, they could get rid of the problem. And they did not take into account that the Staphylococcal enterotoxins are heat stable or very heat stable. So they produced skim milk powder, and of this skim milk powder, or again, milk was produced and offered to the consumer. And as you can see here, for the range or the optimum, and the optimum range for Staphylococcal enterotoxin production, if you look at the conditions we have in milk, which is here marked in green, you can see that we are nearly, except for the temperature, in the optimum range for Staphylococcal enterotoxin production, but also the temperature of 20 to 30 degrees is quite close to the optimum range. So this means that all the conditions were right for the production of the enterotoxins. And so the big problem was, of course, this step, the insufficient cooling, bacterial growth, and the toxin production. And as I mentioned, subsequent heating steps did not affect the produced enterotoxins. And this led to one of the biggest outbreaks with Staphylococcal enterotoxins in Japan. It's about 20 years ago now. And it was in the press everywhere at that time. And here it's said that it has been about 5,000 people affected. The calculations after this outbreak showed that in total, more than 30,000 people have been affected by this case of food poisoning. So fortunately, such cases do not happen every year, but you can see if the conditions are right and there are mistakes made during the production of food, such big cases can occur and cause a lot of trouble, not even for the company, but especially for the consumer. So, thank you very much for your attention. And thank you very much again, Ulash and Kui, for invitation. And to all of the participants, thank you for your participation. And I'm happy to answer questions.
Thank you very much, Professor Dr. Erwin Mauer. You have honored us. I would like to ask maybe Kui would like to say something about us, about you. I'm going to open the voice of the Kui. Kui?
Yeah, hi. Hi, my professor. So I have just one question, because we also focus on the mechanism of the cellulite, how it causes the liver effects by the Bacillus series. So I just want to get more details. Or do you have some new data on the mechanism or mode of action of the cellulite from Bacillus series?
We don't have own data on the mode of action of the cellulite, because, as you know, we more focused in our research on the enterotoxins, which cause diarrhea, and that what I presented is that what has been published during the last years. So
I just wondered. So if we take the, for example, if we have the Bacillus series which can produce cellulite, how it transfer from the gut to liver? So this is always confused me. So do you have any hypothesis or some indicators about this phenomenon?
No, it's a very good question. So, and it's very difficult also, because this point is left out in any of the studies. So nobody really addressed this point so far, and it's a bit against the classical schemes we have from what happens in the intestine, and so in principle it should not enter the blood circulation, but it happens, and it happens also for the Staphylococcal Enterotoxins, which is also quite astonishing. So there must be some mechanisms which we don't know at the moment.
Yeah, I think this is very interesting, but it's also very complicated.
And,
thank you.
It's also a problem. It's not so big as with the Staphylococcal Enterotoxins, but also the animal models for the Hematic toxin are quite rare. So that is also a problem. And as you know, studies with animals become more and more complicated worldwide. So
okay, thank you.
I think we have to live with this not knowledge.
Okay, thanks.
Thank you very much. Thank you very much. And there is also one more question from Dr. Elisha. There is also one more question from Dr. Elisha, and we have opened your voice, so you can address your question.
All right, greetings to everyone, and thanks for the mandate given unto me to ask my question. But let me make this disclaimer, please. I'm an MPhil candidate. I'm not a doctor yet. However, Prof. Erwin, thanks for your marvelous presentation, but there's something baffling me that I want a clarification on. You know, humans, we do take antibiotics as a way of curbing bacterial infections. However, when we consume these food products, which are toxins loaded, for instance, we consume a food product which contains an enterotoxin, okay? Then subsequently, we take an antibiotic. Are these antibiotics capable of curbing these toxins? Because what I do know is they usually target the bacteria and not the toxins.
I'm not sure if I understood the question. So
my question is, when we consume food products containing toxins, for instance, I consume milk, and this milk contains enterotoxin. How do I curb it? How do I control such enterotoxin after consumption?
Okay, thank you very much for your question. He would like to ask, as far as I understand, maybe if somebody consumed the milk containing the enterotoxins, and how can we make the treatment afterwards, or how can we detect the procedure or something like that?
How can we detect the toxin, or can we reduce the toxin, or what was the question? Yeah, or
The detection of the toxin, I think.
yeah, also in milk and in food, Staphylococcal Enterotoxins can easily be detected by these ELISAs.
Mm-hmm.
So they are suitable for analysis of food. So different food. Sometimes if the food has a very high fat content, then sometimes false positive results occur. In this case, you need some kind of sample preparation, but usually all these ELISAs you have seen on the slide are can be used to analyze for Staphylococcal enterotoxins in the food.
Thank you very much, Professor Dr. Erwin Mertelbaumer. As I said at the beginning, it was very special for us, for Kui and for me, that you are here. And thank you very much again for your contributions.
Thank you very much also, Ulash. Thank you, Kui. And you know, it was a pleasure to have both of you in my institute. It's several years ago, but I hope we will meet in presence in the coming years. Thanks again.
We have to meet.
Yes.
Thank you very much again. And the next presentation is going to be held by the Dean of Murcia Veterinary Faculty, Professor Dr. Gaspar Ros. Dear respected professors, we are ready to listen you.
Okay, good morning, and thank you, Lars, for your invitation and for your kind presentation. I'm going to share the presentation, but I need to be habilitated.
Now you can share your presentation.
Okay.
I can open it.
No. Great. Okay, thank you again and for organizing this event. And I'm changing a little bit the topic from Bacillus and Staphylococcus aureus and food safety, which is really interesting. I'm going to present to you some of our research concerning the use of the different, or one particular and powerful antioxidant like hydrotyrosol, which is well known maybe for many people, but since it is not so well known for others, I will begin my presentation talking about this hydrotyrosol. Why are we talking about olive leaves? Well, basically, we talk about these olive leaves because within the European Green Deal is a strategy for Europe to try to optimize the economy based on the agriculture and food chain. It's very important to optimize in different areas of action the economy and the use of resources, particularly the area number 6 from farm to fork, concentrate the interest to obtain different products directly from the production, but at the same time we can obtain from the byproduct or co-product, which is the new method we are using for the leftover of the food industry, trying to obtain the best out of them. And one of the potential and real capacities of this leftover is to extract functional additives. Even though the word additive is not the word that the consumers want to read in the food label, it's part of the components of the food that we want to replace from artificial by natural ones. In this case, from the European Green Deal, we using this byproduct of co-products and the new or already known bioactive components, we can obtain a clean label of food, mean that we can remove the artificial ingredients and incorporate new ingredients free from artificial flavors, color, allergens, products that have to be approved by the European Food Safety Authority, and at the same time have to be listed as an ingredient. So that's why it is an additive, but it's a natural additive. But as a complementary part of this new trend, new strategy, is that not only the use of an additive just replace the additive, it can have, or it should have, an add value. And this add value is concentrated in the action from the nutritional point of view, because we can reduce or remove some ingredient and incorporate new ingredient with potential health benefits. And at the same time, we have to look for this ingredient to be sustainable and with a productive compatibility with the economy. And why are we talking about olive leaves? Because we are talking about also byproducts from the region. I'm from Spain. This is the region where I'm from, the southeast of Spain, on the Mediterranean coast. So here we have a picture with different Mediterranean, as in Turkey and as in other countries in the Mediterranean area, that produce leftover or byproducts or co-products. And one of them, very interesting, is the olive leaves and all the different components that we can extract from them. And one particular and interesting one is hydrotyrosol. This hydrotyrosol is basically a polyphenol. It's within the large and broad spectrum of polyphenol from olive oil, and as well as the fruits and the pomace and the leaves, and that we can obtain from the olive tree. This hydrotyrosol is particularly interesting because it has been demonstrated that it's a very natural, powerful antioxidant. It's 10 times more antioxidant and with high power scavenging activity if we compare with other polyphenols that are used as a reference. For instance, green tea is 10 times more antioxidant or twice the Q10 enzyme, coenzyme. So this is very important, as well as that this and the hydrotyrosol can be obtained from different roots from the olive fruit, but not so much that if we compare with the olive leaves. However, it is important to know that we have to know in advance, we have to characterize the byproducts or co-products, because it is strongly affected by cultivar and the environmental conditions. So it's not the same from one region to other, cultivar, climate, irrigation, so on, so on, so on. And that is really a key point in the food industry, how to optimize and use the best or the most adequate. The southeast of Spain is, of course, well known by the olive oil and the olive trees as well, and of course all the byproduct that is obtained from this industry. And the region of where we are located in the southeast is very active in the fine chemistry, the chemistry that can be used, and clean chemistry to obtain this natural extract from the byproducts. If we concentrate our interest in this polyphenol, the most important is oleuperine, but oleuperine can be broken down into hydrotyrosol. And as you can see in this image, I go back a little bit. In this image, these are different well-known and potential activities like anti-platelet aggregation, anti-hypertensive, anti-inflammatory, anti-atherosclerosis, antioxidants, and as well another potential activities to prevent dyslipidemia. Within this wide range of potential effects, we concentrate only in two main areas so far. One is the antioxidant capacity to preserve and increase the shelf life of the meat product. At the same time, if we can add some anti-inflammatory benefit with the consumption of this concentrate. See, from olive leaves, we can obtain quite easily different staphylococcus with water, supercritical extracts, and so on, hydrotyrosol, because it is breakdown, as I mentioned, from oleuperine alicon, and from here you obtain hydrotyrosol and hyaluronic acid. All of them are understudied. But something important with the hydrotyrosol is that the concentration in the final extract is not so high. It depends, as we mentioned before, different parameters from the product, but at the same time, at the end, you can obtain only a quite low concentration, 8% in power or 25% in serum. But there's some fine chemical procedure to obtain synthetic. I want you to bear in mind this idea that we have natural, not so high hydrotyrosol, and synthetic. We can obtain synthetic from 20%, but it's important to know that you can even reach almost 98% of this hydrotyrosol. This product is water soluble, and the main problem we have been detected with this hydrotyrosol is the bitter taste, because it is responsible for the taste and intense taste in some olive varieties of this bitter taste that is concentrate when you obtain this extract. That's why it's important to remove all the bitter components, so to obtain a bitter-less or no bitter product. Of course, something important, it has to be approved by EFSA and FDA and produced in Europe. And in my case, this hydrotyrosol was validated by EFSA as a novel food to be used as an ingredient in different sources, because this hydrotyrosol was validated as a safe for the proposed post-consumption specification, batch-to-batch variability, stability, and production process by one company from Murcia. So this company reached EFSA and demonstrated that these are synthetic hydrotyrosol can be safe. And in fact, this is one of the different brands. It has been used for fish, vegetable, margarines, and we've taken in or bear in mind also the level of safety for this, the use of this hydrotyrosol we have applied into meat. And with this meat, we try to improve the quality, at least maintain as an strategy at the same time to add some health benefits. Why meat? Because meat is supposedly within the Mediterranean pyramid is not in the bottom, it's in the top. It means that we have to consume meat on a weekly basis. I mean, not every day, only once a week, maybe twice a week. And if it's red meat or pink meat, it's not a debate, or processed meat, less than that. But the fact is that in Europe, although many, the behavior of the consumer is changing, and now people eat less meat, mainly young generation, but still the consumption is quite high. It's the main course daily in many houses. This is the trend in Spain, where 40% of the Spanish population has reduced or not tried to red meat. But the point at the same time is that they are concerned about what they eat, and what they eat at the same time has to be on what they label, what is in the label that on the product that they purchase. In fact, this is in the trend in Europe as well in the United States. People are looking in this label because they read it, but they don't understand many times that this ingredient has to be natural or organic, and mainly in meat, not only the origin, but at the same time, the ingredient. That's why there's some different strategy that has been in place, implemented in both in the fresh meat and in the meat product to obtain new meat with a clean label and at the same time with health benefit. This is a strategy that we are following the mark in this red arrow. We are adding antioxidant and polyphenols, even though there is a wide range or option to obtain this healthy product from farm to the consumer. Just before I present to you some of our experience with this hydrotyrosol, this clean label in meat industry means that the ingredient has to be natural obtained, no E number, which is critical, that this E number can be removed, no number, but just the name of the natural product. The problem is that the ingredient in this case are not so well recognized, and at the same time, the consumer, for a clean label, pretend to have a product with natural or minimal process and contain less or no additive or preservative. And of course, all of them has to be simple product with a simple label to be understood. How antioxidant and the hydrotyrosol in meat products, how can act? Okay, this is part of our research since 2015. We concentrate to try to understand how can we use this hydrotyrosol if it is compatible with natural ingredients. So we have studied meat products from poultry, from lamb, and from pork, basically. This is our first publication, the effects of hydrotyrosol, combining. This is the first one of strategy, is to introduce olive oil as a natural salt combined with walnuts, high in polysaccharide fatty acid, and a small amount of hydrotyrosol. This is how it was designed, this study. And in this case, we only use one concentration of hydrotyrosol. Remember, this is natural. This is just for olive oil water, clean extraction, with a concentration of 50 ppm. In this case, we produce chicken frankfurter, and this is how it was mixed with walnut and extra virgin olive oil. And in fact, what we try to understand is if this extract, natural extract, bitterness, can be compatible or can be has the same effect that walnut or olive oil, virgin olive oil. From the nutritional point of view, we obtain a product with no differences, only mineral difference has some small differences, and the fatty acid profile as well. But our main interest was to obtain product with the same profile and to identify sensory analysis. We detect this hydrotyrosol. And well, our surprise is that the product were very acceptable by the panelists, and in some cases, we found that the panelists even gave a higher score to these products with a hydrotyrosol. So it is more or less for the consumer close the idea of this hydrotyrosol to the olive oil, which is much natural, of course, but at the same time expensive for the food industry. Then what we do was to try to compare with different concentrations from 23, 7 and 7% from different origin, water and leaves and straight water, the same concentration of hydrotyrosol, the same product. And in this case, we concentrate a little bit more into the color, lipid oxidation, and protein oxidation as well as the scanning electron microscopy and sensory analysis. And we found that the hydrotyrosol from leaves were acceptable on the other cases. We made some comparison with mineral because in the first preliminary study we had some impact on mineral, but we really found that there was a real impact on this mineral viability, taking iron, zinc, and selenium as an indicator, mainly zinc and selenium, because the main objectivities project in this case was to try to increase this natural component in the skin to make it more resistant to the process. It was a challenge that we achieved, and at the same time we try to analyze, but really hydrotyrosol has very little impact on the mineral absorption. As I mentioned before, then we went to compare natural hydrotyrosol versus the synthetic one, and in this case we prefer to use a different food matrix. We use lamb meat because it's the meat with the highest content in fat, more saturated, and that's why we compare in this case this synthetic and natural hydrotyrosol in this land. But at the same time, since we are in the Mediterranean, one of the areas of research is not only olive byproduct, but at the same time rosemary. Rosemary is also very powerful. It's a reference as a natural ingredient, clean label in many meat industries, and the idea was to try to replace this rosemary for hydrotyrosol. And we found that this could be a good source, the synthetic, because it's even more powerful to preserve color, oxidation, and at the same time to have an antimicrobial effect. So synthetic hydrotyrosol is one of the potential one. The problem is the price is so expensive, and the companies that produce this synthetic hydrotyrosol are so limited that some of them went into the bankrupt. In the region that they produce this synthetic is because the food company cannot afford to include this synthetic. So we remain working with this olive leaf hydrotyrosol. And how can we obtain not only antioxidant, but at the same time anti-inflammatory or health benefit? Then we organize one study. In this case, we concentrate into the study, in this case, with pork meat, particular ham, pork meat, because with this ham we have been working before, and we obtain a low salt and reducing salt and low fat and unsaturated fatty acid with a processing, and we study the anti-inflammatory and antioxidant effect. But in this case, we concentrate in a preclinical study, which main objective was to study the reformulate cooked meat, in this case ham, using the mice C57BL6 studies with mice for 2 months consumption, and during 1 month they were obtained fat animals with a cafeteria diet, and then we incorporate, in this case, one control, one obese, one with bioactive directly, one with a ham reformulate, and one with a ham as a control. So we compare these 5 groups. Well, this is us 2 times. The first time is to induce the obesity, and the second time was to see the impact if we can reduce this obesity. The trend on the biomarkers is that those in green, mainly in the reformulated ham when they consume, anti-inflammatory in the intestine in the body, were potentially favorable. Sorry, in Spanish, I know the copy in English. And with this, we can clarify that there's some mechanisms that may lead to a claim in these products. This is just a potential effect. As well, we demonstrate this in the liver, the fat depositing liver that were really reduced with the obese and the obese reformulated ham. This is the spec of result where after 1 month from the obese animal, they reduce about 50% this fatness, so can be included, we can have some potential benefit. This is a scanning spec of the fat is in yellow. How can you remodulate after longer period, more than 1 month? So finally, this is my conclusion, is that the green labeling of meat products is a growing trend that has to be addressed by the meat industry to fulfill the consumer demand and expectation. It does need clear definition, legal offering, labeling for an adequate and fair use of natural ingredients by the food industry, and consumer has to be taught about the real meaning of this clear and simplified labeling. The benefit is an open to discussion by the food industry, scientists, and consumers. A natural plant extract can add some additional value to meat product, but it has to be tailored for each meat product. It's not the same for all of them. It's a process that takes time and obtains solid scientific evidence. Claims are not just a marketing tool. My final remark is for 2 of the latest open papers that you can read about hydrotyrosol with all these elements combined. And this is just my recognition to the team with the food industries and with my colleagues. Thank you very much for your attention, and I am open to any question or comment regarding this topic.
Thank you very much, dear professor, for your valuable presentations. If there is any question and we would like to have them, you can write them in the chat box, or you can click on the reaction sections, and then we can open your voice for addressing your question. If you have more questions, you can send also the email to us, and we can address it to Professor Dr. Kasper Ross. Dear Professor, thank you very much for your valuable time. It was very nice that you are here.
Thank you very much, and have a fruitful Congress. And I will be open to answer any question if you receive. I will send you the presentation so students or attendant can take the reference of the papers to read. And if they have any question, please feel free to contact me.
Okay, may I give you your email address on this chat box, and they can write to you?
Yes, please. Feel free.
Thank you very much, dear Professor.
Thank you. Bye-bye.
Bye-bye. The next presentation is going to be held by Professor Dr. Zara Mosley. She is from North Macedonia, from Kirill and Methodius University, Faculty of Veterinary Medicine. Dear Professor, thank you very much for accepting our offer, and we are happy to listen to you.
Good morning to everyone, to every participant in this Congress. First of all, I would like to thank you, Professor Ulaş Açaros and Damla, for inviting me to be part of this very well organized Congress. So I would like to open my presentation here.
Firstly, you can open your presentation. Then afterwards, from the bottom green section, you can share it.
Yes, I know. Wait a minute, please.
Yeah.
Yes, the presentation is open it. Share screen. Do you see my presentation?
Yes, we can see.
Okay. So for today, my presentation will be for multi-class, multi-residual LC-MS method for veterinary drug residues, mycotoxins, and pesticides in urine. First of all, I would like to say that the part of this
Dear Professor, are you there? Or there is a problem with your internet connection?
Development, optimization, and validation of solid phase extraction method for multi-class simple preparation, mostly in urine. After that, development, optimization, and validation of liquid chromatography tandem mass spectrometry method for determination of veterinary drug residues, organophosphorine pesticides, and mycotoxins in urine. And finally, after the development and validation of this method, the application on multi-residual analysis as a pilot study in terms of using of the method suitability. As we know, today in a global marketplace, food safety and food quality have gained increased attention from mainly food consumers, food producers, and governments. Veterinary drugs, such as different class of antibiotics, are widely used in food producing animals as
I think there is a small internet connection problem, Professor Dr. Zara Mosley. I'm contacting her now. Dear respected participants, there was an internet connection problem of Professor Dr. Zehra Hayrullahi Musliu. She's trying to reconnect again. I will request a few minutes of you. Dear respected participants, we need a few minutes till she get the internet connections. During meanwhile, we will open music. Dear respected participants, it seems it will take a while for fixing the internet problem, and that's why I would like to ask Professor Dr. Hedayet Hosseini for starting his presentation. He is from Iran, from Shah Beteshi University of Medical Sciences. Dear respected Professor, thank you very much for accepting of our offer for the presentation. It is nice that you are here. I'm allowing you now for sharing.
Hello, everyone. I would like to thank you, Professor Ulaş, and the other organizer of this Congress, and also executive committee for establish this Congress, this useful Congress, and thank you for inviting me to have opportunity to present my presentation in this Congress. If you let me, I would like to share my presentation.
Now it is open. You can share, dear Professor.
Yeah, thank you. Okay.
Firstly, you have to open your PowerPoint.
Yes, I did it, and thank you.
Now we can see.
Yes, you can see my presentation. Thank you again for inviting me. My presentation is about effect of curing agent and cooking on biologically active polyamines level in turkey meat, and I would like to introduce the importance of polyamines and its effect on human health and consumer. As you know, polyamines are nitrogenous compounds that naturally exist in meat, in fish, for example, cheese, wine, and in lowest level in other foodstuffs. This group of compounds mainly includes putrescine, spermidine, and spermine. The main compounds are these, and these are synthesized endogenously in the body or made by microbiota, and also obtained by diet. And as you see the figures of these compounds, these are, as I told, nitrogenous compound, and these are a little bit similar. And this is the cycle of the production of these biologically amines. This cycle starts from ornithine. As you know, ornithine is an amino acid in the meat, for example, or turkey or poultry tissues. It starts by decarboxylation reaction. Ornithine by enzyme ornithine decarboxylase start this procedure, and the final product would be putrescine, and also putrescine start to change to spermidine and spermine in the cycle of biosynthesis. As you see in this figure, these products are changed to each other, and also, as you know, these are spoilage indicator. As you know, putrescine, spermidine, and aspermine have very bad smell, and these are spoilage indicators in meat. And when meat goes to decay situation, these bad smell products make it undesirable for consumer. Also, in regard of health and safety aspects, cell growth and differentiation is one of the polyamines biological role in the human body. However, ingestion of this compound can accelerate the growth of tumor cells and cancer. And it's the main concern about use these compounds from meat and other sources, for example, cheese and wine and other foods I mentioned before. Therefore, decrease in their concentration in food product, particularly in meat and meat product, are crucial and important for human health. In this regard, the reduction in the level of polyamines, mainly aspermine, in the daily diet is suggested by experts in cancer prevention and therapy. As you know, curing is a very important and crucial section and procedure in meat industry to improve quality of products. And the most of meat products have this procedure for high quality and improve taste, color, smell, and other textural or visual aspects in the meat processing industry. During curing, mainly salts, for example, sodium chloride, nitrate, phosphates, and acid ascorbic are generally used to increase shelf life, eating quality of meat products, and these additives influence the microbial load, pH, lipid oxidation, water holding capacity, texture, color, and flavor. And most of sensory attributes of meat products is made by these additives and curing procedure. One of the most important compounds we use in curing is salt, which is seasoning, help digestion, and also preserve and flavor enhancer. There are many roles for salt in meat products. Also, nitrate influence both taste, color, and microbial load of turkey meat product. As well as you know, acid ascorbic helps nitrate, for example, sodium nitrate, and its chemical ingredients help to accelerate curing process and the nitrate processing aid. To reduce the polyamines concentration in food matrices, there are many ways and many suggestions. One of them is about the additive in formulation research. Other researcher finds salt has a great influence on biogenic amines and reduce the biogenic amines. Sodium nitrate also and nitrate and nitrite also have a good impact on decrease polyamines. As you know, these compounds are used to preserve the meat and meat products and have a destructive effect on microbial load as well as in polyamines. Some other techniques, for example, vacuum packaging, modified atmosphere packaging, and also other method of cooking depicted, they are important and can decrease the polyamines in meat and meat products and make the level of these compounds less and less in procedure. In this study, we evaluated the concurrent and interactive effect of regular meat curing additives and cooking method on production of putrescine and aspermine, which are these two compounds are the most important biogenic amines in this regard. And mainly we had three main goals. The first goal was to find the effect of each curing additives on polyamines in torquemides. The second goal was to find the interaction and concurrent effect of these additives in potassium and spermine level. And the third goal was to find the optimum concentration of additives to find the lowest level of polyamines in Turkey cured meat products. For this research, we purchased fresh turkey meat from market in Tehran, and we cut it into cube shapes from breast of turkey meat, and the size of the cuts was about 50 gram. In the clean situation, we tried not to add any additional microbia to these parts cut of turkey meats. Then we did curing. Sample were immersed in the curing solution containing design amount of salt, sodium nitrate, sodium polyphosphate, and ascorbic acid. This suggestion gets from design of expert software and suggested runs performed in this study, which I will show you in table 1. And for curing, a sample were kept in glass container in refrigerator at 4 degrees of centigrade for 6 hours. And then we cook the meat, turkey meats, cured turkey meats. For cooking, we divided the samples into 2 parts. One part endure boiling cooking. We used boiling water for 30 minutes, and for frying, we immersed the samples in 120 degree of centigrade canola oil for 10 minutes, and for each side of the meat samples, we used 5 minutes, and totally we used 10 minutes for frying the turkey meat samples in boiling. We used response surface methodology, RSM, in this research and control composite design for designing the study. And the treatments which we did in this study was suggested by this software and the methodology of central composite design in different level of salt, sodium chloride, sodium nitrate, sodium polyphosphate, and ascorbic acid, which these were independent variables. And also the other independent variable was cooking method, which, as I told, was boiling or frying. Based on central composite design, which, as I told, suggested by expert design software, 4 numerical category of variables defined, and also 1 non-numerical variable defined in this research. In table 1, you can see the variables, independent variables, and the range we used in this study. As you know, as you see, salt per gram, it was from 0 to 2% in curing methodology. Sodium nitrate used from 0 to 200 ppm part per million. which it is the highest level permitted in standards. Sodium polyphosphate was from 0 to 0.5%, which also it is the highest limit permitted in standards. And ascorbic acid also, the range per million. And also, as I told, these 4 variable are numerical, and the categorical variable was the way of cooking, which divided into shape, boiling and frying. Then after curing and cooking, we tried to measure polyamines in samples. As I told, 16 runs were suggested by the software, and we tried to measure polyamines in these samples. And for increase the velocity and efficiency of the extraction, sample were microwaved at 500 megahertz for 2 minutes. Afterward, sample were cooled and the mixture transferred into centrifuge tube and mixed with 2 ml Aureus solution and centrifuged at 1252 G for 10 minutes at room temperature. And then we did or performed the HPLC analysis, high performance liquid chromatography instrument used in this study. 510 microliter of extract solution, which contains 450 acetonitrile and 60 microliter octanol. And also 2 grams of salt were added to the prepared sample and then shaken vigorously. Then the sample were centrifuged, as I told, at the same situation, and 20 microliter of supernatant were collected and injected to HPLC with UV visible detector. And the condition defined for the HPLC was the failure rate about 0.8 milliliter per minute. For mobile files, as I told, acetonitrile and water, and detector was set up at 254 nanometer. And also the separation performed ODS column of HPLC. After performing this method, we were able to report the spermine and putrescine amount. As you see, it's a chromatogram which we get from HPLC instrument. The first peak is belonging to putrescine, and the second peak is for spermine, as I told, after analyzed but by HPLC UV methodology. As I told, our first goal was about the effect, to find the effect of different compounds used for curing in this research. And as you will see in table 2, for spermine, based on central composite design methodology, we would be able to predict the responses given two different variables. As I told, we had 4 variables: sodium chloride, sodium nitrite, sodium polyphosphate, and ascorbic acid, and also the categorical variables, frying and boiling. And based on the finding, we found different compounds had different effect on amount of polyamines, spermine in meat samples. In this table, in table 2, you are able to see the variables, sodium chloride, nitrate, sodium polyphosphate, and vitamin C, and cooking method. And the sum of squares, which calculated by the software based on the amount of measures we performed by HPLC, you can find the effect of each compound on spermine concentration in turkey meat cured and cooked in this research. And the most important compound had effect on spermine concentration is nitrate. As you know, the amount of this sum of squares is highest, then it has more effect or impact on the reduction of polyamines of spermine. The second score is belong to sodium chloride for spermine amount in poultry, in turkey meat, cured meat, and the second score is belong to sodium polyphosphate, and the last one is for vitamin C. Vitamin C or ascorbic acid had the lowest amount effect or impact on the reduction of spermine in samples, and as you see here, nitrate had the most important effect on the amount of spermine in the samples. And as you know here, cooking had not any effect on the spermine amount. And for putrescine, also we did analyzed, and also we used software for calculation and to find the most important and the lowest important effect we can view in table 3 for putrescine analysis. As you know here, it is similar to spermine findings. The first score is belong to nitrate, sodium nitrate. It had the highest sum of the squares and shows it's the important factor for reduction putrescine in turkey cured meat. The second score is belong to sodium chloride, and the next one was sodium polyphosphate, and the last one was vitamin C. As I told, it was completely similar to finding about spermine, and the impact of different variables you are able to see was the same in this regard. And also for cooking, we find not significant effect for reduction of putrescine in cured and cooked poultry and turkey meats. As I told, sodium nitrate was the most effective additive, plays an important role in polyamines content in cured products, and it was matched and agreed to other researcher reports. And sodium nitrate demonstrated the most important role in the inhibition and the reduction of the polyamines, and it would be for inhibit the growth of amine producing bacteria in meat during curing time. Also, it's demonstrated that salt decrease the polyamines, as you saw in the table 1 and 2. Salt had the second score in reduction of polyamines, putrescine and spermine. This result also reported by other researchers in this regard, and the reason why salt caused to decrease polyamines is related to unique properties of salt in food, sodium chloride, spermine or potassium, and also have obvious impact on enzyme and water activity of animal tissue, which inhibit production of these polyamines, biological active amines. Sodium polyphosphate was another variable in this study. It was in third role. The role of the sodium polyphosphate was in third score. As you know, sodium polyphosphate have a good impact in curing for water holding capacity, increase pH, and control lipid oxidation, and helping other agents to lessen polyamines level. Though this study about the effect of sodium polyphosphates on polyamines are rare, or finding shows it causes mild reduction in polyamines amount. And also the acid ascorbic is a common additive used in curing. Although acid ascorbic showed positive effect in controlling lipid oxidation and also in meat quality in other research, but seemed it has no outstanding influence on reduction potassium and spermine content in cured turkey meat. And last factor was thermal as reducing factor 2 and lead to decrease in polyamines content. However, contrast results were reported in this regard, or finding depicted there is no any significant change in terms of applying culinary treatment, which is presumed heating effect are covered by other strong agent like sodium nitrate. Our second goal in this study was to find the interaction between salt, sodium nitrate, sodium polyphosphate, and other additives concurrently on the biogenic amines level in meat and in turkey meat. Three-dimensional surface of interactive effect of salt and sodium polyphosphate is depicted in figure 3. We will see this figure. It showed that when amount of polyphosphate and salt increase, a significant decrease in polyamine content is seen, and it should be noted that cooking was in average mode, and sodium nitrate and ascorbic acid was adjusted constant in this section. In figure 3, you can see the three-dimensional graph of interaction between polyphosphate and sodium chloride. As you see here, when the amount of sodium chloride increase and when concurrently polyphosphate amount increase in curing, the level of polyamines, spermine and potassium decrease, and we found synergic effect between sodium chloride and polyphosphate. Also, we studied the interaction effect of sodium nitrate and sodium polyphosphate in spermine and in potassium, which illustrated in figure 4 in next slide. That it revealed that incorporation of sodium nitrate into sample in the presence of sodium polyphosphate could reduce spermine content dramatically, and the effect and impact of this synergy was higher than before treatment. As you see in this figure, three-dimensional graph you see here, when the amount of sodium nitrate decrease in the amount, and when you see the polyphosphate amount increase, the level of polyamines, potassium and spermine decrease dramatically, and the impact of decrease of polyamine when we studied the concurrent effect of nitrate, sodium nitrate, and polyphosphate was noticeable. And the last and third goal of our study was optimization of curing agent concentration for having the less amount of putrescine and spermine in cured samples of turkey meat. Based on our finding and applying software prediction methodology, the optimum amount of salt, sodium nitrate, sodium polyphosphate, and ascorbic acid to minimize spermine and putrescine content was 2 grams sodium chloride. The optimum level, the optimum level of ingredient was 2 grams sodium chloride, 200 ppm sodium nitrate, 0.5 gram sodium polyphosphate, and 382 ppm of ascorbic acid. In this level of additives for curing, we found the less amount of spermine and putrescine in turkey meat samples. And the validation amount was high, 0.963, which showed it was completely in good situation for prediction. And in this figure, in figure 5, you see Ramp's view of the optimization curing. Also, you see again the amount of sodium chloride in 2 gram per 100 gram, nitrate 200 ppm, sodium polyphosphate 0.0, and also vitamin C 382. And you see the cooking method. It was categorical, not numerical variable. And you see the method of cooking had not effect on the spermine and putrescine amount. And you see the less level of the spermine is here in this level of the additives, and also for putrescine we found the lowest level in in this situation. Okay, in conclusion, we found sodium nitrate, sodium chloride, and sodium polyphosphate are strong agent in decreasing polyamines level in turkey meat curing. However, the increased role was seen in ascorbic acid. Moreover, cooking methods were not as powerful as curing process in polyamine reduction. Also, synergic effect was observed between sodium nitrate, sodium chloride, and sodium polyphosphate in curing process. And the optimum amount of salt was determined regarding to the minimum level of polyamines content in turkey meat for protection consumer health. Thank you for your attention, and I'm ready and open if there is any question, or if more necessary description is necessary in this regard. Thank you again, Professor Ulaş, and all participants for attention my presentation. Thank you.
Dear Professor Dr. Hidayet Hosni, thank you very much for your nice presentation. Is there any question to the presentation of Professor Dr. Hidayet Hosni? Maybe if we are allowed, we can put your email address on the chat box, then they can direct the questions to you by emailing you.
Sure, thank you. I will write my email address in the chat box, Professor.
Thank you very much, dear Professor, for your contribution.
Thank you again.
The next presentation is going to be held by Zara Mosley. I think she has fixed her internet problem.
Yes. Did you hear me? But I can't start video.
If you can start the presentation, then it is not problem, dear Professor.
Okay. Did you see my presentation?
Still not. First, you have to open your PPT on the.
Yes, the PPT is open now.
Then from the bottom side, there is a green
share
button.
screen.
Yes.
And I'm sharing now. Did you see?
Now we can see.
Oh.
It is open now.
It is open?
Yes, it is open now.
Okay, thank you. So I'm so sorry for the disturbing you, and I'm so sorry for the changing of agenda for today.
You are welcome, dear professor.
Okay, for the today I will present multi-class multi-residue LC-MS method for the veterinary drug residues, mycotoxins, and pesticides in urine. As Professor Ulash said, I am from Faculty of Veterinary Medicine, and I am head of the Analytical Laboratory for Food and Feed Analysis. This presentation or this study is the part of the internal atomic energy project, which is funded, and the title of the project is Integrated Radiometric and Complementary Techniques for Mixed Contaminants and Residues in Foods. What are the specific objectives of this study? First of all is development and optimization and validation of solid fast extraction method for multi-class and sample preparation in urine matrix. After that, development, optimization, and validation of liquid chromatography tandem mass spectrometry method for determination of veterinary drug residues, organophosphorine pesticides, and mycotoxins in urine. And finally, after the validation of the method, application of the method as a pilot study in terms of testing of method suitability. As we know, in the global marketplace, food quality and safety have gained great attention, mainly from food consumers, food producers, and governments. For that reason, on the other hand, we know that the veterinary drugs, such as different class of antibiotics, are widely used and administered in fruit-producing animals to prevent or to treat disease. Also, some veterinary drugs like anabolic hormones, lactones, beta agonists, thermostats, and show growth-promoting effects and are commonly used for these purposes. But on the other hand, also, we know animals are often exposed to mycotoxin mixtures along with other contaminants such as heavy metals and pesticides, making multi-residual and multi-toxin exposure study relevant from a public health perspective. We know the development of analytical method for the determination of residues and contaminants in food of animal origin plays a key role in the protection of public health. But there are a large number of analytical methods for determination of maybe one class of veterinary drug residues or only veterinary drug residues. But there are a very small number of methods which are multi-class and multi-residuals, which analyzed both veterinary drug residues and contaminants in one method. So most of these methods, as I said, have been developed for the analysis of each group of residues and contaminants, but they are only separately. For example, we have methods only for determination of different class of veterinary drug residues, or we have methods for determination of pesticides or for determination of mycotoxins, but there are very rare methods which cover both and drug residues and mycotoxins and pesticides in one method. Before 2 years ago, I have the similar presentation, which was held in Antalya in Food Congress. I was presented the similar method for multiple residues contaminants, but in bovine meat. As you can see, we have published the paper. So we have one which is in press, the similar method, but the matrix is different in milk, and today's presentation is in urine. So the aim of this presentation is develop an analytical method for the sensitive determination of 72 multi-class determinations of antimicrobials, thermostats, anabolic hormones, lactones, beta agonists, mycotoxins, and organochlorine pesticides in bovine urine using liquid chromatography tandem mass spectrometry method. The second aim of this study was optimization of MS-MS method, investigation and optimization of extraction procedures, and in the end, after the optimization of each step, we followed with the validation of the method. So in this study were included as I said, 72 compounds, from which 42 were veterinary drug residues, from which 4 were thermostats, 6 anabolic hormones, 2 lactones, 10 beta agonists, 15 antibiotics, and 5 sulfonamides. After that, 28 organophosphorus pesticides from contaminants and 2 mycotoxins. Also, in this study, for better quantification of each analyte which were included, were added 17 isotopically labeled internal standards. As I said, were included thermostats, anabolic hormones, lactones, beta agonists, antibiotics, sulfonamides, organophosphorin pesticides, and mycotoxins. And from isotopic labeled standards, internal standard, as you can see, were included approximately 17 isotopic labeled standards, which were purchased also from International Atomic Agency. So, simple preparation. In the first step, 30 milliliters of urine samples were centrifuged for 5 minutes, and after that, only 5 milliliters of urine samples was fortified with the standards and internal standards as working solutions. In the next step, 5 milliliters of sodium acetate buffer and 5 milliliters of phosphate buffer were added, and the mixture was shaken 1 minute in a vortex. After that, after cooling at room temperature, samples were centrifuged 5 minutes at room temperature. After that, the next step was enzymatic hydrolysis, which was incubated 70 hours at 37 degrees. And after cooling in room temperature, the next step was centrifugation, 5 minutes at room temperature. Before the extraction in the Oasis HLB cartridges, we know that the cartridges first they need to activate it and coordinate it. The activation was done with 5 milliliter methanol and 5 milliliter water, and the whole volume of 10 milliliter was passed from cartridges, which cartridges passing was 1 drop per second. After that, the cartridges were dried, washed with 5 milliliters of water and dried again before elution step. The next step was elution with 2 different elution mixtures, elution mixture 1 and which followed with 4 milliliters of elution mixture 2. Elution mixture 1 was consist from methanol, ethyl acetate, and ammonium hydroxide elution mixture 1, and elution mixture was consist from methanol and dichloromethane. After that, the step was evaporation and underwater bath at 35 degrees in the stream of nitrogen near to dryness. The residues was reconstituted with 1 milliliter of mobile phase, which mobile phase, as you can see, is in a 95 with 5 volume mobile phase and mobile phase A and B. After that, the residues was filtered through a membrane filter and into auto-sampler wheels prior to liquid MS analysis. As I said, the main purposes of this study was not only development of multi-residual and the multi-class method, also was and validation of this developed method. So, as the validation parameters for quantitative purposes were linearity, limit of detection, quantification, decision limit, detection capability, accuracy, which was evaluated through recovery, and precision, which were evaluated from determination of repeatability and reproducibility. First of all, we needed to do the optimization of MS method. Why? Because the high selectivity also of the MS system, which can overcome problems related to the presence of interfering compounds extracted from really complex matrices such as urine in this study, qualitative and quantitative analysis are severely affected by the unwelcome matrix effect, such as proteins, salts, which interfere with analytes of our interest, mainly when we are using electrospray ionization, both in positive and negative mode. So, as I said before, we have used for MS operation, electrospray ionization were evaluated for determination of all 72 compounds, as I said, in both positive and negative ion modes. In this study, all 72 analytes were measured in MRM mode. According to the European Regulation 2002/657 requirements, the confirmatory accreditation for substances in foodstuffs as target analysis requires a minimum 3 identification points. We monitored 1 precursor ion and 2 product ions, providing as well 4 identification points which fulfill European Union requirements. After the optimization of MS method, we followed with the optimization of a mobile phase. Mobile phase compositions and gradients are very important consideration in liquid chromatography method, particularly for trace analysis, in order to achieve high sensitivity, good ionization, and sufficient separation with minimum interference from matrix. When I say particularly for trace analysis, we know from the European regulation the substances of group A, we have minimum required performance levels, which are the banned substances, and we need to have a very sensitive method for their detection. So we tried, as you can see from the slide, 6 different mobile poise, and the end we find the optimum mobile phase, which was water with 5 millimole ammonium acetate, 0.0% formic acid, and 0.01% trichloroacetic acid, while mobile phase B was methanol with 0.1% formic acid, which we gained improved separation, good peak shape, high signal sensitivity, and finally the best peak symmetry and resolution. While the other tested mobile phases, we gained poor separation, bad peak shape, low signal intensity. We have detected only one daughter ion, which followed with parent ion, and unfortunately, some compounds were not detected, as you can see from the slide. From metocryphos and cephaloxins, we gained only one daughter ion. So other chromatographic conditions, as you can see from the slide, were flow rate 0.2 milliliter per minute, injection volume 10 microliters, column temperature was 40 degrees, and the run time was about 20 minutes. After the optimization of MS method, optimization of liquid chromatography, the next aim was the optimization of the extraction procedure. We tried 4 extraction protocols which were spaced at 3 concentration levels. So, as you can see from the slide, we tried 2 methods with a liquid extraction, which one was with enzymatic hydrolysis, which tyrostats unfortunately were not detected or extracted. Also, we tried liquid extraction without enzymatic hydrolysis. Also, tyrostats were not extracted. After that, we tried extraction with a solid phase extraction method without enzymatic hydrolysis, which we gained low recovery, less than 35% for anabolic steroids and zeranol. Unfortunately, stanozolol and tylenol were not detected, while solid phase extraction with enzymatic hydrolysis, we gained very good recoveries. As you can see from the slide, the best recoveries was gained from the solid phase extraction with enzymatic hydrolysis, and recoveries were between 65 to 115%, which fulfilled the European requirements according to 657/2002 regulation. The other extraction protocols with liquid extraction together, both with and without enzymatic hydrolysis, we gained very poor recoveries between 40.3 to 77.7%, while solid fat, other methods, solid phase extraction without enzymatic hydrolysis, we gained also very poor recoveries from 47.3 to 101.5%. Results and discussion of the validation study. So, to test the linearity of the method, matrix match calibration Curves were prepared on each validation day to give 5-point calibration curves around and ranging from 0 to 100 microgram per liter. As you can see, linearity was acceptable for all compounds which are analyzed. The determination coefficient was more than 0.99. In the validation parameters, we have limit of detection and limit of quantification. So 20 duplicated negative samples per matrix were selected and spiked with the standard solution. Then after that, they are pretreated and analyzed according to the method which we described above. So the limit of detections were calculated from the lower concentration of the analyte, or I can say the lower standard which we used for calibration curve, and as a mean value plus 3.3 times of the calculated standard deviation. While for the limit of quantification, this was plus 10 times of the calculated standard deviations. As you can see, limit of detections were very low, 0.07 microgram per liter for beta agonists, mainly from brombuterol, malbuterol, till to 5.551 microgram per liter for metacriphos. While limit of the quantification, the lower limit was 0.19 microgram per liter, also for brombuterol, and the maximum was for metacriphos 16.7 microgram per liter. Decision limit and detection capability, which can comply with the requirements of European Decision 2002/657. Also, they are calculated because in the study were included, and mycotoxins were calculated according to DJ Sanko 2004 to the regulation is to 1007 to 6. In the case of substances for which no permitted limit decision was obtained, mainly for substances of group A, which they have minimum requirement performance levels, the decision limit was obtained by fortification on 18 blank sample standards at the recommended concentration level in equidistant steps. So CC alpha was calculated from the corresponding concentration at the U-intercept plus 2.33 times the standard deviation within the laboratory. Sorry. While the detection capability, or CC beta, was obtained by analyzing 18 blank samples fortified with the analyzed at the decision limit and calculated as corresponding concentration at the decision limit plus 1.64 times the standard deviation of the within laboratory reproducibility, also according to the European requirement 6/7 regulation. So CC alpha was between 0.11 microgram per liter for clenbuterol to 10.88 microgram per liter for cefalpirin, while CC beta detection capability was lower for beta agonist, 0.15 microgram per liter for clenbuterol and maximum 15.23 microgram per liter for tillozin, which were according and in terms of European Decision 657/2002. Accuracy was evaluated also from recovery, as you can see from the slide, in 3 different concentration with a 6 replication per level, and the results was minimum for marbeterol 65%, spiked at concentration, lower concentration 0.2 microgram per liter, while maximum was for brombuterol 115%, which also was spiked at concentration 0.2 and 0.4 level. Precision, intraday precision or repeatability was calculated with intraday precision, also was calculated in 3 different concentration with 6 replicates. And were evaluated by preparing, as I said, 3 different concentration levels. But here I have to mention for substances which have minimum requirement performance levels at 1, 1.5, and 2 times, while the substances which have maximum residual levels at 0.5, 1, and 1.5 times FMRL, using, as I said, 6 replicates for each concentration level in 1 day. The results are for licomycin 1.26 spiked at 10 microgram per liter, while maximum was for malathion 23.31% spiked at 10 microgram per liter, which also covers the European regulation, while because the lower concentration when we use the coefficient of variation did not exceed more than 20%. Precision, which was evaluated from interday precision or reproducibility, were evaluated by analysis of spiked samples at the same concentration levels on 3 consecutive days, also in 6 replicates for each concentration level. Coefficient of variation as reproducibility for lincomycin was 2.29% spiked at 10 microgram per liter, while maximum, which except the European regulation, 29.42, carbaryl spiked at 10 microgram per liter. And the conclusions were, the method describes extraction, cleanup, identification, and quantification of 72 residues of veterinary drugs and other contaminants in bovine urine. This study included optimization of MSMS method, optimization of liquid chromatography method, optimization of extraction procedure, validation study, isotopic label standard, internal standard for better quantification. We have monitoring and distribution of residues contaminants in the extraction procedure, quantification of compounds, which I mentioned before, using the internal isotopic label standards, and in the end, I can say the method could be used in routine analysis of bovine urine samples for simultaneous detection of veterinary drug residues and contaminants, including pesticides and mycotoxins. That is all. Thank you for the listening.
Dear Professor Dr. Zara Mosley, thank you very much for your valuable contribution and nice presentation.
Thank you.
Is there any question to the presentation of Professor Dr. Zehra? You can also write your questions in the chat box, and we can direct the questions to the professors.
Okay. I
Thank
am here to answer for every question.
If we are allowed, we can put your also email address on the chat box, and they can write to you also directly.
Thank you.
Thank you very much, dear professor, for your valuable contributions.
Thank you. Thank you too.
Dear respected participants, next presentation is belong to Dr. Stefan Stangagiu. He is expert in the apitherapy and a very famous person in the apitherapy area. And he was honorable president of Generally Apitherapy Association, and now he is Romanian Apitherapy Association head. And at the same time, he is the general secretary of International Apitherapy Federation. Welcome, Dr. Stefan Stangagiu. Thank you very much that you are here, and we are happy to listen to you.
Thank you very, very much for the invitation, Professor Ulash. Thank you very much for all of you coming here. It's so fantastic to see so many people from all over the world, top scientists, very, very good people. So I have a bit of heart, palpitations. How can I convince these wonderful, so intelligent people that propolis is important, that they should use propolis every day for their patients, for their family? So I'll do my best. And now, Professor Ulash, how much time I have? 20 minutes, 30 minutes?
20 to 30. We have 30 minutes.
Okay. So I have prepared my PowerPoint. I will go a bit quickly through the slides, but I'm sure Professor Ulash will save these videos. And then later on, you can stop the video anytime and look to the graphics and so on. Okay, so let me share the screen first. And let me change a bit here the type of the setting. Now, wait a second, as I see here. Yeah, I want to take this out. Okay, so, okay. Let me know if you can. Now I'll go to full screen. Can you see the full screen?
Yes, we can see.
Okay, thank you very much. So I'll start. Okay, so propolis is used for thousands of years, knowing or not knowing about its properties. There are papers which says that propolis was used in ancient Egypt, in India, in China, almost all over the world where it has been wild bees, but also where it has been beekeepers. So, and propolis is so famous because it has so complex composition, very, very useful substances, especially polyphenols. And the bees knows that the trees, the buds, they protect themselves, like you see here, against radiation, against bacteria, against viruses, against parasites, against mechanical stresses and thermical stresses with these substances. So the bees, they collect these substances. The bees are very clever, and then they bring to the beehive. And the scientists, they found out that these very good substances can be extracted. And there are several kinds of extractions, several types of propolis. You see here different colors. And what is amazing is that propolis, these substances, are known by the plants, by the insects, but also by the human beings and by the mammals. So the living cells, they are okay with this kind of substances, which has practically organic acids, polyphenols, minerals, and so on. So we can even inject propolis in various concentrations inside our body. And as you'll see, I'll explain a bit later, also intravenous. A few guidelines about this topic. I'll go quickly through original types, importance of life, for life on Earth, using the beehive, characteristics, composition, properties, indications, administration. It's like a sort of a very intensive course on propolis. Origin and types, we see here European kind of propolis, here is Brazilian kind of propolis, Bacillus dracunculifolia, it's the tree here, and it's another one, Dalbergia castophyllum, both in Brazil, but also we find Dalbergia in many other countries, all tropical countries. Okay, so the color, you see the form, it's like this one, and different colors, but still composition, even if the composition is different, we have many similar properties. There are differences, but there are also many common properties. You see here about the extraction of propolis in this type, water propolis extract or alcoholic extract. And now a few words about the geographical and botanical origin. So you see here in this table made by Professor Vasia Bankova many years ago. We find propolis in all continents, in Europe, America, New Zealand, Brazil, Russia, Cuba, Sicily, all over the world, all continents, we can find these plants which are producing the polyphenols which are collected by the bees. Now you see here the propolis type like poplar, green, birch, red propolis mediterranean, Clusia pacific, and so on. And you see here the main constituents. And what is interesting, even if the composition, the constituents are different, they have, as I said, common properties like antibacterial or antiviral, which means that propolis is oriented towards the function. So the function of propolis is more important than the composition. Final function is more important than the composition. Okay, so we know that the plants, they are protecting themselves against bacteria, parasites, and so on, ultraviolets with these polyphenols, I told you before. If the plants wouldn't have these polyphenols, the sunlight, the ultraviolets, the infrared, and so on, the cosmic radiation would destroy the plants. So no plants, it will be very difficult for us to live on Earth. Of course, we can get good foods and good medicine also from the oceans, but on the continents it would be very difficult to live without these polyphenols. So the plants, the living plants, they are constantly attacked by bacteria, by viruses, by fungus, by all kind of microorganisms which wants to eat or to use the energy and the substances from the plants. So the plants, they must defend themselves. So they create these polyphenols, and we can say again that no polyphenols, no plant's life, no plants, no bees, because the bees, they need the fruits, they need the flowers, the nectar, the pollen, these protective substances. So no plants, no bees, and no bees, we can say also no propolis, because the bees are collecting this fantastic propolis and they are bringing it to the beehive, and then we can extract it through several methods used by the beekeepers and by the scientists. Also no propolis, no bee life, and I'd like to tell you a few words here why the bees are collecting this propolis, these substances, because they want to protect their own life cycle. Because at all of these levels, almost larva level, pupa, and so on, adult, the bees are constantly attacked by this aggressor, chemical aggressor, biological aggressor, physical aggressors, and so on. So they need protection. And for them, the best protector is propolis, because as you'll see, propolis has many, many different properties, useful properties. Here is one example in beekeeping. The beekeepers understood that when a beehive, a bee colony has a lot of propolis, the colony is healthier, it's better, more active, less diseases. So now many beekeepers, they advise the other beekeepers to use this kind of wood, which has many, like this kind of, I don't know how to tell, this scratches on the wood, which makes the bees to put much more propolis. When the wood is like this one, is very, very light, it's very soft, the bees will not put so much propolis here. So less propolis means less protection for the colony, including against some diseases. But also no bee life, we can say also no human life, because the bees, they are pollinating thousands and thousands of flowers of trees and shrubs and plants and so on, which are ensuring the biodiversity. But also they are making all kinds of fruits and seeds, which are very important for our life. If we look, unfortunately, our civilization is nowadays going through self-extinction. This idea, this concept with profit, it's killing us. What we are doing for the planet, it's like we are, I can say, even if it's very, very tough, but we are like the cancer for our planet. We must change these systems. Industrial agriculture is a disaster for life on Earth. Pollution is a disaster for life on Earth. So we should not do such things. We should plant thousands of trees. And by the way, I stimulate all of you on 26th of March. Next month, it will be a global event in the world. We'll plant thousands and thousands of plants. And please join us on this initiative launched by Slovenia. So the bees are using propolis in the beehive for many, many things, to glue the frames, to block the entrance of the enemies like mice or other insects and so on, to have an air inside, a healthy air, which is also taken in beehive air therapy, like these aerosols from the bees, from the beehive, antiviral and so on. So I go through some other ideas about the plant sources. Okay, then preparations on propolis. We have many kinds of preparations and products, over 30 different preparations. Most of them, they start from this kind of extracts: water extract, alcohol extract, or hydroalcoholic extract, or oil extract. And there are some other ones also, like butter extract, like olive oil extract, like essential oils extract, and even in the seeds and in the pollen. Okay, so propolis is not completely diluted in only alcohol or only water or only seeds. So you need a mixture of several solvents to get almost everything from propolis. Here is one example with the alcohol extract of propolis, and this is water propolis extract from Brazilian propolis. So you see Brazilian propolis is much more water extractable. And this one is more like gluish. And this is very good for the skin, for mucosas all over the body. But when you want to enter inside, in between the cells or in the cells, the water extract is a bit better. It's better. Now, the pH value of propolis is a bit acidic. So this is important when you give propolis to children which have some bacterial infections, or to adults. Do not mix propolis in milk. Some others, they say, oh, let's put the propolis in milk because the children, they like milk. But when you put in milk, milk is more towards alkaline, towards this more pH, higher pH. So the acidic antibacterial and antiviral substances from propolis will be more or less blocked. So better is to get the propolis in honey and then take it. There are over 300 different main substances in propolis. We can speak 1 month about this composition of propolis. It's fascinating. And I wrote a book several years ago on propolis, and I found over 70 useful properties in propolis. I'll tell you only some of them. Antioxidant. We know that oxidation is a big danger for our health. If we have antioxidants, we can protect better our body, inside our body, outside our body. We know that oxidation, free radicals are causing all kinds of diseases in the body. And we know also that propolis, the plants are using these substances in nature against the fire, against the heat, against destruction, against the free radicals. For example, in Australia, when the forests with eucalyptus are burned, then these buds, these seeds, they resist against the fire because they contain such kind of substances. So it is very, very important, this property of propolis, because with antioxidant, we can prevent hundreds of different diseases. Now here, we have also antioxidants in the fruits. We see here the polyphenols, and again, other properties: antibacterial, antiviral, antimycotic, antiparasitic. You see here this image. And then anti-inflammatory, as I told you, immune modulator. This is very important, especially now, because we are still in COVID time, and we know that sometimes the virus creates too strong immune reaction, like the cytokine storm. But propolis is immune modulator. It can calm down the immune system if it's too strong. or it can increase the health, the strength of the immune system when it's too weak. It's the modulation kind of effect, which is to be found also in case of honey and other bee products. So the immune system cells, when they have the tissues, the organs, but also the cells, and all these cells, they need all kinds of protection, and propolis can help a lot here. Another property, cicatrizing, in wound healing, in anti-tumoral. Like here you see pinosembrin, one of the substances having these properties, including local analgesic, galangin, other properties. Quercetin, which is finally recommended against COVID, it's also useful. Caffeic acid phenethylester, other properties, caffeic acid, hepatoprotector. There are thousands of articles on propolis. Just go to PubMed and you'll find now over 3,600 articles on propolis, related to propolis. Here's some mechanism of action of propolis against bacteria. So you can notice here that propolis does not work against bacteria with only one mechanism or with only one substance, like the classical antibiotics, the pharma industry driven antibiotics is acting, but propolis acts through many different simultaneous, I repeat, simultaneous actions against the bacteria. And this is very, very important. Now, in case of antimicrobial effect of propolis, you can use it on many different kinds of disease and problems like you see here. There are papers, very good papers presented recently in the last years by very good scientists, which shows also the mechanism of action of propolis against the pathogens. Okay. Some words about indications, contraindications of propolis. Because propolis has natural substances, and these natural substances are from the category of the good substances. They are not toxic substances. We can use even huge amounts of propolis every day, like 70 grams of propolis, which is a huge dose of propolis. We can take every day and will not have toxic effects. So the body accepts very much. So that is why we can use it against thousands of different conditions and diseases, but there are also some contraindications. So first of all, let me show you some photos about the use of propolis in medicine. Here is a tree, and this is the skin of the tree, if you want, is the bark of the tree. And when a tree has a wound, the tree will secrete these substances, which are antibacterial, antiviral, antiparasitic, and they are also cicatrizing. They are wound healer. And the same substances which are collected by the bees, they can be used also not for the vegetal tissue, but for the human tissue or for the animal tissue. And it has the same effects, cicatrizing, wound healing. Here is, again, this kind of photo, like we can use honey and propolis and combinations inside the wounds directly. Here is another example from Italy, from Dr. Francesco Ferraboli, one patient having a big infection in the bones. The infections in the bones, when the patient is also diabetic or has a low immune system, cannot be cured. And because the immune system is too low, the blood flow is too low, but propolis can help in this situation. You see here, they use gentamicin even inside the bones, but no results. And then finally, 2 times per day, propolis inside the bones. It was propolis tincture, alcoholic extract of propolis inside the bones, destroyed the bacteria, and then the recovery was in 30 days. The wound was closed in only 30 days. And there are other examples here, like in case of big accidents, like this part of the finger was cut, an accident, and then we can put propolis directly, and in a shorter time, like less than 1 month, it was complete cure and close the wound. And you see here also later the result. In dentistry, it's used also in several countries, the propolis, in Romania, in Uruguay, in England, in Great Britain, in Germany. Many countries, they are using propolis because propolis is against the pains. It's more or less a painkiller, not very strong like the anesthetics, but it is still very good. Then it's anti-inflammatory and antibacterial. And because of this, propolis can help a lot the dentist to have a more relaxed life, no fear. Another use of propolis is for the to prevent periodontitis and parodontosis later, because propolis, it's very glue-like, it's very adherent. It stays on the teeth like here for hours or even days. So when you need it, when you have some tooth-related problem or gum-related problem, the propolis will stay much longer than the other medicines. So this can help to lose, help against the losing of the teeth. And here is Dr. Rafael Felitti from Montevideo, Uruguay. He comes to many of our conferences. He's a dentist there, and he used propolis and honey extensively on his patients. And you see some of the photos he sent us. The injection here in the tooth, directly to the tooth canal of propolis tincture, 20%, acting as anti-inflammatory, antibiotic, and analgesic. And you see here injections until this area, and with very, very good results in a couple of weeks. Then also on the skin, we can use this. Is a doctor from Bulgaria, Plamenenchev. Many kind of such problems can be cured with propolis, especially if it is used before some honey to extract the dead tissues, the dead cells, and the microbes. Then propolis works faster. Then against traumas, traumatic lesions on the body. Here is against diabetic created wounds like gangrene, diabetic gangrene. You see here in less than 3 months, combination with honey and essential oils. Another very interesting application is to put propolis on the breathing masks, because when you have the propolis here, you breathe an air which is having also anti-inflammatory, antioxidant effects. And some people, it's some studies like in Egypt, they said that this propolis is also antibacterial for the lungs. Okay, so there's a special technique. You need to blow the air from the lungs, but blocking the nose in the same time. And this will open the lungs, the bronchi better, and then it's good to inhale the propolis. Then the propolis will go deeper into the lungs if you use this kind of method. Then we know that in the last about 20 years, it's a fascinating other way of using the bee products, beehive air. And the most important group of substances in the beehive air comes from propolis again. And but not only propolis, also the beeswax, which is giving the larvae, the pollen, the bee bread, the honey. Even venom has volatile components which comes into the beehive air, and thus the children, the adults can use it for prevention and treatment of many diseases. Okay, so here is one such bee house in Germany. We specialized in beehive air therapy, very, very clean. Contraindications, as I said, only a few of them. In case of allergy, some people, they are allergic to propolis or the substances from propolis. Intolerance, this happens especially for people which are dehydrated, which have a dry mouth, or people which are having a lot of sensitivities and other allergies. Blood coagulation, propolis is blocking, it's healing very quickly the wounds, the superficial wounds especially, but also it improves the blood fluidity. So when you give some anticoagulants, together with propolis, you must be a bit cautious to check the fluidity of the blood with your specialist in hematology. Blood pressure, in some people, propolis, because it's antispastic, it may decrease the blood pressure. So people having a low blood pressure should be cautious when taking propolis. And also organs transplantation, because propolis is immune modulator, but has also this immune enhancement property. And when somebody has a transplant organ, like a liver transplant, then the immune system of the patient must be kept under control at least a couple of years. And then later on, very, very small doses can be added to the treatment of the patient to improve the overall functioning of the body. But of course, under the coordination of a good immunologist and the team which made the transplant. Okay, so one golden rule in administration of propolis is to use the propolis to administer it through several anatomical ways to better reach the target. So not some people, they take only through the mouth, only through the nose, or only through the skin, but you'll see that it's much better to use all anatomical ways, because if you use all anatomical ways, including intravenous, there is one experimental study now in Germany and also in China, they do it for a long time with propolis extract, water propolis extract in the veins. But the most common one is oral administration, sublingual administration. Now in COVID times, it's wonderful as aerosols. And also, as I said, skin, but also as suppositories, not only for the children, but also for the adults. What is the best dose for propolis? The best dose of propolis can never be said in advance. You must make tests. You must increase the dose gradually until you reach the best effect. And this is okay in chronic diseases, because you have time to do it slowly and to see what is the best doses for you. Once the patient reached the best doses for him or her, then they should stay on this dose, like healing dose, until the doctor, the specialist said, okay, now you are cured, you are improved, everything is now good. So you can diminish the dose afterward, but not to zero. This is the case, recommendation in case of drugs, chemical drugs, medications, chemical medications from pharma industry. They said, okay, now you can stop, go to zero, because the chemical drugs, they have a lot of adverse reactions. But propolis in our situation and many other big products like you see here, after you use them as healers, like medicinal drugs, they can continue to help us. We can continue to take them with some breaks, but as a prevention dose, as preventative remedies. And we take them in this situation like foods or like food supplements or like, you know, this kind of alicaments, how we said sometimes. But in case of an acute disease like a burn, we should use higher doses much, much quicker. We do not have time to make correct allergy test or tolerance test. But the risk of getting infections after a big burn or an accident or something like this is much bigger than getting some allergies. So in this situation, you go very quickly at high doses. And you can block the bad effects, for example, of a burn, because you can block very quickly the free radicals and you can restart the regeneration. So the idea is to go through all anatomical ways to reach all cells in the body. And all these anatomical doors or ways. And here I have some images, like we can use also propolis in eye drops. There are some companies which are making these eye drops with propolis or with royal jelly, creams, and as I said, oral administration, then semi-liquids. We can use also like honey and propolis mixtures. We can use as solids, raw propolis powder, granules, butter, propolis, propolis tablets, bonbons, capsules, microencapsulated propolis extract, or as toothpaste, chewing gum, water extract, tincture, raw propolis with pollen, oleaginous seeds, crystallized honey, beeswax. This one is a very, very interesting method because we can get a lot of active substances without the need of alcohol. So when somebody does not want to use alcohol for various reasons, they can take raw propolis as fresh as possible from a beekeeper, and they can chew it. They can suck it in the mouth for maybe 1 hour, and then they can chew it with this pollen or oleaginous seeds. And then propolis will break in billions of small particles, so the surface contact will increase a lot. And then they can swallow, and then they will release a lot of active components which are good for our health. So propolis is also good for pharynx. Like in case of COVID-19, we know that the virus attacks the nose first, but also the pharynx next one. So we can do gargling with propolis and honey. And some people, they have sore throat. So we do this kind of gargling and also spraying the throat. And in case of bronchitis or coughing, we can also use the bee products, especially propolis, through inhalations or nebulization. Okay, now a very, very important element here to understand how propolis works, and honey too. We know that when we breathe, we inhale a lot of dust, a lot of particles, we inhale also viruses and spores and so on. So all these unwanted substances, particles, they are eliminated from the lungs by these specialized cells, the cilia, through this mucus. And these cells, they are also under constant attack by the free radicals. For example, if somebody is smoking or somebody stays near a smoker, will inhale the smoke, and the smoke attacks the cells. So if we can inhale some small amounts of propolis, this propolis will help the cells against these free radicals. So they'll be able to work better to eliminate faster these particles, including viruses. Okay. Now, other administration to the mask, like I said, as suppositories here, or nebulization. This is a very, very useful method now against the respiratory viral diseases. Okay, combination of heat and propolis, they are propolizer devices made in Italy, for example, like these ones. You just heat the propolis to about 80, 90 degrees Celsius, and then propolis becomes more liquid and then becomes volatile, and then it can be breathed. And then you can put it in your living room, in bedroom, and you can inhale it normally through normal respiration through the night or through the day. Or a very, very famous way of using propolis is through in the cars. There is this special device. You just put it into this part of the car here where you get heat. Some people which are smoking the car, they are using this special part of the car. But you can use it for good things, not for cigars. Use it for propolis. And then your car will become like a sauna, like a propolis sauna, more or less. And then it can act like a treatment room. Believe me, it works wonders. And then another device is made in Slovenia. There are more and more countries now which are doing such kind of devices, also in Germany. And it's the same effect, the same idea to propolize the air from the room where you stay. Another administration, suppositories, very effective for children, but also for the adults, because it goes into the bloodstream by bypassing the liver. Then other kind of administrations you see here for eye drops, as I said, ocular drug films, molecular mycelia. Okay, for the skin, cream, sunscreen agent, plaster lotions, and so on and so on. As soaps, in cosmetics, it is used also for the hygiene of our body. And for tissues also, we can administer propolis through electroenphonophoresis, sometimes when you need deeper in the body, like in a joint, or ionotherapy. And also we can combine with other remedies, other products like honey or ginseng. Okay, sublingual administration. I go a bit quickly here. Okay, through massage, you can use a propolis cream and you can apply it to make massage. You can also apply it and stimulate the acupuncture meridians or acupuncture points, depending on the diagnostic. Okay. Okay, here acupressure, like one of the techniques I told you before. One very, very good product which is made in Turkey is this propolis made by Balparmak. It's an extract of propolis. It has no alcohol inside, but it has a lot of active components inside. So it's very practical to be taken under the tongue. Okay, so also to treat a lot of diseases. And I arrived to the conclusions. What are the main needs of our over 100 trillion cells to be happy? All our cells and tissues and organs in the body, they need these categories of things. And usually in medicine, we are in a hurry many times, and maybe we speak a bit with the patients about nutrition, but not so much. We forget to speak about hygiene. We forget to speak about repairs, regeneration, reconstruction. And we forget also to speak about relaxation. And if it's too much stress, too much activity, not enough relaxation, and so on, at cellular tissular level, we cannot heal a patient. So every single category from this graphic here is very, very important if we want to get healing. Same apply for all cells in the body. This category applies for all cells and tissues in the body, including for the immune system. And the immune system cells, they need all those categories I told you before, because there is another thing with when we have all these categories, we can make happy all these big organs. And there is a cooperation between the organs, like it is the cooperation between the cells. Like the pancreas helps the immune system, but also the immune system helps the pancreas. So it's a bilateral cooperation between all our organs. And that is why many people are dying still because of COVID, because they have a viral infection, which attacks the lungs and then the immune system. But they have many other problems in the body, like cardiovascular problem, like high blood pressure. They have diabetes, or they have other conditions, and they are killing the patients mainly, and not so much the virus itself. So you need to have an integrative concept, integrative thinking, and make happy all these organs. Okay, and now one other idea, the propolis has hundreds of different substances, as I said. Antibiotics or antivirals, they have only one molecule. So what do you think? What is better, to have an army of different components or just one molecule? Of course, it's logical, only one molecule, it's not a good idea, it's not a good strategy. You can get some good results for some time, but the bacteria, the viruses will learn how to defend against this one molecule system. But the bacteria, the virus cannot defend against an army of different substances. That is why Mother Nature, that is why the trees are on Earth for millions and millions and millions of years. And we know that bacteria and viruses are older than the trees, but still the trees, they survived because they found ways to keep under control these bacteria and viruses. So, and this, the secret of the trees is multiple substances, not mono substances. And finally, if the bacteria would be a terrorist, would you fight against a terrorist with only one weapon? Will you use only one molecule or more molecules? Will you use only one antibiotic? Will you use this kind of thing? Can you fight the terrorists with this kind of thing? Antibiotic is like this guy, using just an arch. And this is not very clever. So we need to use multiple weapons, like in the armies are using against terrorists multiple weapons, not only one weapon. And the same is like these weapons in our case in biochemistry, in biology, in apitherapy. These are our weapons: polyphenols, essential oils, terpenoids, aromatic substances, and so on. And because these substances are destroying or keeping under control the bad pathogens, And you find, as I said, thousands of articles on this mechanism of action and how they work. Here, just a synthesis of propolis properties. Some of them, I told you, there are over 70 such properties. One of them here, chelation of heavy metals. Just think to the bad effects of the heavy metals in the body, like mercury, which is causing multiple sclerosis and so on. So we can have all these good properties by using propolis. And conclusion, in order to obtain fast and good results in our practice, we need to ensure the following conditions related to propolis: perfect quality, which means fresh, raw propolis, correctly used or extracted. An old propolis is not very good, it's oxidized, it has less aromatic substances. A fresh one is much better. So you need to cooperate very well with your local beekeepers and kindly ask them to give you very, very fresh propolis immediately after they collect it. And of course, the quality is very, very important, and the quantity too, as I said, in the doses. Then we need to use various pharmaceutical forms like raw tinctures, hydroalcoholic creams, all kinds of extracts, suppositories, and so on. We need to administer the propolis through all anatomical ways, as I said, because we'll make happy all the cells in the body. Do not use only one form. Try to use all anatomical ways, and you'll heal faster and better. And you must use it at the right time. Like, for example, if you want to prevent COVID, you need to take it before you go into the public, before you get a risk to get the virus. So in the right time, in the first symptoms, you must act very, very quickly. And you need to use it until complete cure or clear improvement. Do not take it only 1-2 days. Take it until you are completely cured. Final conclusions. Propolis is an excellent protector, a biological body guard, a good friend for humans and animals, health, vitality, beauty, and longevity. If you want more information about this topic, please feel free to write me an email to my email address, or you can also Google my name, Stefan Stangasiu, or also you can send me a WhatsApp message to this number. And do not forget to visit our website, apithera.org, which is the website of our International Apitherapy Federation. Thank you very, very much for your attention. And thank you again for your participation to this event.
And thank you very much, Dr. Stefan Stangacı, for your valuable presentation. It was very nice to listen to you. Is there any question to the Dr. Stefan Stangacı? You can write in the chat box, or if you click on the reaction sections, we can give you if you are allowed, we can write down your email address in the chat box, and then we can resend the questions to you directly. Thank you very much again for your valuable. In the meanwhile, I would like to announce here, we are now in the preparation with Dr. Stefan Stangacı to organize an international congress about apparent therapy in the middle of the June. I would like to invite also interested person in this event from here. And thank you very much again, Dr. Stefan Stangacı.
Thank you. Thank you.
And the next presentation is going to be held by Dr. Ahmed Ali. He is from India, from Mumbai University, from the Department of Life Sciences. Dear Dr. Ahmed Ali, we are happy to listen to you. He is here, but I'm just trying to contact to him by WhatsApp. I will request just 1 minute from you. He is here. He is here. Okay, dear doctor.
Very good afternoon to all of you from India. Yeah, I hope I'm audible. Can you confirm it's am I audible clearly?
We can hear you, but there is some noises, extra noises. Maybe
Yeah, just a minute. I'll just, yeah, just a minute. Okay, I hope it is clear now.
Okay, we are listening to you.
Yes, very good afternoon, and it's been long awaited meeting all of us after almost a year. We had the same event almost a year in February, and as Dr. Stephen mentioned, this time crowd is much bigger and we have a better audience, so it gives kind of like a nervousness to all of us to speak and deliver our lecture without any wasting, because the session has been very wonderful. So I'll not take much of the time. I'll just quickly go to my presentation. Before that, I'm thankful to Dr. Ulas and his team, especially Dr. Damla, for giving me this opportunity to be with in the panel of esteemed scientists. So we had a very wonderful lecture from Dr. Stefan about propolis, and he has rightly justified that this propolis can be used for various purposes in our life. I'll be talking about a work which we are doing. So I'll just share my screen. So is my PPT visible, Dr. Ulas?
Yes, we can see. We can see.
Okay, so I have made it full screen. Yes, thank you again, Dr. Ulas and your team for making this wonderful arrangement by bringing us, and hope this the next edition will be physical, where we can meet and interact with each other. So my topic will be sugar toxicity and its consequences, hyperglycemia and diabetes. So I'll be talking basically about the biomolecular events which happen during this process, which we call as glycation, and what we are working as anti-glycating agent. So the overview will be like, I'll very briefly talk about diabetes and its relation with glycation, the theme of my presentation, and what are these diverse molecules which are known as advanced glycation products. And I'll also give a brief of the molecules which we are exploring as anti-glycating agents. So diabetes, one of the major non-communicable disease, has become a major public health concern globally, which has led to an increase in the number of people affected by this disease, and ultimately causing severe cost on the health system of governments. So NCD risk factor collaboration came with the data that there has been a 4-fold increase in the number of diabetes from 1980 to 2014. And earlier it was supposed to be a disease of the developed countries, but if we analyze the data, we can see that half of them are living in developing countries also, like India, Brazil, Indonesia, apart from USA and China. So if we can see that this graph shows that the population of diabetics will be around 650 million or more than that by the end of 2045. So it's growing. There are several factors because of that. I'll not go into detail. Lifestyle and food habits and all these things are there. So the diabetes can be identified because of the major symptoms, which we know that hyperglycemia and hyperlipidemia. These conditions induce inflammatory responses due to glucose oxidation, induction of polyol pathway, synthesis of prostanoids, and glycation of proteins. And all these processes, they further lead to a situation that is known as oxidative stress because of the generation of reactive oxygen species. And this process also adversely affects the antioxidant defense system. So as I mentioned that all types of diabetes have common features like hyperglycemia and glucotoxicity. This accumulation of glucose leads to an increased amount of generation of undesired products in the body. So accordingly, the modern theory of diabetes suggests that the oxidative stress and glycation are main causal factor of the diabetes and the secondary complications of diabetes. So this chart, this graph shows that when there is an increase in the concentration of glucose, the high condition is hyperglycemia. The proteins are worse affected, and this glucose causes different types of modification to the proteins directly or indirectly. One of the major protein modification mechanism is glycation, wherein there is a non-enzymatic interaction between the sugars and the proteins, and the interaction is between the carbonyl and amino group respectively with all the nitrogenous compounds. So this process is different from the one process which we see, enzyme regulated process, which happens in the endoplasmic reticulum and Golgi apparatus. That process is known as glycosylation. The adversity or deleteriousness of glycation is also because of the irreversible nature of this process. So you can see this diagram. You have we have proteins in the body, in the circulation. They are encountered with the sugars, and the interaction between the sugar, especially glucose, and amino group of the proteins leads to the formation of safe bases. And then we see the generation of early glycation products, and then they get converted to advanced glycation products, and they finally modify the proteins. So the process of glycation can be divided into 3 stages: early, intermediate, and late. In the early stages, the reaction starts with the interaction between the aldehyde groups of glucose and amino group of epsilon amino group of lysine, arginine, or N-terminal amino groups of the proteins, leading to the formation of CIF bases. This reaction is very fast. So these CIF base adducts or unstable products, they undergo chemical rearrangements slowly to form early glycation products. So you can see the representation of here you can see sugar and protein interact, lead to the formation of CIF bases, and then to the Amadori products, which are called as early glycation products. Then we have dicarbonyl compounds, and then advanced glycation end products. So in intermediate and late stages, these CIF bases and Amadori products undergo various kind of chemical rearrangements like oxidation, reduction, and hydration, and ultimately leading to the aggregation or cross-linking of the proteins. So once the process crosses intermediate stage, the reaction becomes irreversible, and the reaction is also slow. It takes several days or weeks or almost a month or something like that to show its consequences. So final brownish product which is generated is known as advanced glycation products. When I started the lecture, I said that diabetes is identified by hyperglycemia, and then we talked about the glycation or the consequences of that high concentration of glucose. But it does not mean that glycation does not occur at normoglycemic level. It happens at normoglycemic level, but the body has a mechanism to take care of those glycation products, especially advanced, early and advanced glycation. They are neutralized by the physiological mechanisms which we have in the body. So the damage is caused under excess or high concentration of glucose, or the damage is realized more under hyperglycemic conditions because of the high amount of advanced glycation products generated in the body and their adverse effects. So advanced glycation products are basically a very heterogeneous group of molecules. They are generated through different routes. One is auto-oxidation of sugars, Amadori rearrangements, and CIF bases. We also have ingested food as exogenous source of AGEs in the body. This is realized when we bake the foods. The browning happens of the cooked material. So that is also a very good source of advanced glycation products in the body. So as far as their classification is concerned, AGEs can be defined as or can be categorized as cross-linking and fluorescence. They can be fluorescent, non-fluorescent, or cross-linking. So some well-known age markers are carboxymethyl lysine, carboxyethyl lysine, pyrrolene, pentosidenine, etc. Now, what are the factors which affect age production? These are oxygen-reactive oxygen species and redox active transmitters. So formation of AGEs during these conditions. Environmental factors like diet and smoking also influence the production of AGEs. For common mass, one of the very good glycated product which is seen in the lab test is glycated hemoglobin, which is denoted as HbA1c. It accumulates in the body as a result of diabetes, and it has been used to know the history of diabetes because the level of HbA1c gives an idea of glycemic history of the previous 120 days. So it has been used, it has been accepted to monitor the patients with diabetes. We can see here. So glycated hemoglobin has improper function as compared to the normal hemoglobin. Now, what happens when there is glycation in the body? We see several consequences: aging, diabetes mellitus, Alzheimer's, Parkinson's, cataracts, retinopathy, arteriosclerosis, and increased susceptibility to infection. These are some of the pathophysiological conditions which arise due to the glycation. At molecular level, the toxicity of glycation is realized through the inhibition of biomolecules, and the processes which cause the impairment or inhibition of the function of biomolecules include precipitation of proteins, cross-linking of proteins, aggregation of proteins, ROS and RNS generation, strand breakage of DNA. So we can see that these processes are involved in any of our biochemical events. Body has a mechanism to detoxify the glycation products. We have enzyme systems like aldehyde reductase, glyoxylase, amadoreases. We have proteasome systems which degrade age-modified proteins. There is a involvement of age receptor which is called as rases. We have macrophage system which dispose the peptides. The one drug which has been approved is aminoguanidine, which has been shown to have its own side effects. People suggest that, or researchers have been suggesting that vitamins are quite potent enough in protecting from the menace of glycation, especially the generation of free radicals. And we have anti-glycagenic agents and antioxidants from the plants. So I'll be just explaining what we have done so far with two molecules. One is a natural product obtained from Nigella sativa, that is thymoquinone, and the another version is a very commonly used artificial sweetener, that asulfame potassium. So these are the assays which we have done: fructosamine and measurement of early and advanced glycation products by fructosamine and carbonyl content. We have used a spectrophlorimeter to measure the total ages. We have used the thioflavin T and Congo rate based method to analyze the aggregation of the proteins which is induced by glycation. We have also used electrophotic methods to see the impairment of biomolecular structure, and we have done interaction studies. So this is the molecule thymoquinone. So as you can see from this compiled graph, that thymoquinone is has a very good potential in inhibiting the advanced glycation product, that is measured in terms of carbonyl content, that is by more than 59.3%. Similarly, there is a significant reduction in the early glycation products, which corresponds to almost the similar amount of advanced glycation products. Browning, measurement of browning is not a very reliable method, because sometimes browning is not observed. But it is an indicator that glycation may have happened, which was discovered long back in 1912, and it is also known in food industry as Maillard reaction. So when we analyze the glycation-induced aggregation of the BSA, we could show that there is a severe reduction, almost by more than 40% in case in the amylide cross beta structure of the glycated sample by the thymoquinone. When we measure the amount of total ages, you can see that BSA plus glucose combination gave a very high amount of advanced glycation products. When we added thymoquinone in the increasing concentration, we could show that at higher concentration, at 20 micromolar, there was a almost reduction by almost 80% in the amount of total ages. But these total ages, the name reflects it's these are only fluorescent ages which are measured by fluorimetry. Here we measured that we analyzed the fluorescence spectrum, where we analyzed the quenching of the BSA by thymoquinone, and we could show that there is increased quenching with increasing concentration of thymoquinone. That clearly showed that there is an interaction between the thymoquinone and BSA. Similarly, we did a secondary structure analysis, because glycation affects the secondary structure of the protein, especially alpha helical and beta plated sheets. So we could show that glycation impairs the structure of secondary structure of the protein, which was maintained or which was not allowed in the presence of thymoquinone. So it means that the secondary glycation-induced secondary structure alteration is also prevented by thymoquinone. Then we analyzed the glycoxidative damage of DNA. The DNA is damaged because of glycation adducts as well as reactive oxygen species which are generated in the late stages of glycation. We could show that we use the plasmid PBr322. We could show that this is the control DNA, and the second lane has the DNA which has been damaged due to the glycation system. And we have used lysine, methyl glycol, and FeCl3 as the glycation system. We could clearly show that there is a severe damage to the supercoiled DNA, and most of the DNA is now in the form of circular DNA. Thymoquinone had no effect on its own. On the other hand, in lane 4, we can show that the presence of thymoquinone prevented the glycoxidative damage of the DNA. So then we also analyzed the significance of asulfame potassium. Actually, we wanted to check what kind of effect this artificial sweeteners have on the glycation process. We were actually expecting that they will be also involved in glycation process, but their involvement was a very surprising result for us. We could see that if the presence of asulfame potassium could severely affect the browning and generation of early as well as advanced glycation products in the in vitro system. We did congruent and thioflavin T assay to analyze the aggregation of the proteins. So we could show that the aggregation of the glycation-induced aggregation of the BSA is prevented in the presence of asulfame potassium. Total ages was total fluorescence ages was also prevented in the presence of asulfame potassium. So as I mentioned in one of the studies, like in case of thymoquinone, we could show that secondary structural alteration takes place in the presence of glycation. We did SDS page analysis to see whether there is any change in the secondary structure, and in turn there is any change in the migration pattern. So the lane 1 shows the BSA, native BSA, which is not treated with the glycation, or that is not treated with the glucose. Lane 2, you can see that there is aggregation of the proteins. That's why we could not, here we are not able to see the monomers and dimers clearly. And there is formation of oligomers of the protein, which can be seen near the wells. Presence of asulfame potassium prevented the aggregation of the proteins, which we could confirm through this STS page also. Then we check the role of this asulfame in the glycation-induced damage, that is glycoxidation damage, or DNA, glycoxidative damage of DNA. We can show here that lane 5 shows the lane 4 shows the reversal of the DNA damage, and lane 2 shows the damage of the DNA. We did the fluorescence emission spectrum. We could show that there is increased quenching with respect to increased concentration of asulfame potassium. So that shows that there is an interaction between the BSA and asulfame potassium. This is a very important parameter to claim that a molecule which is for which we are checking the bioactivity can be used as a drug. Then we did the transmission electron microscopy to analyze the size of aggregated particles. You can see this in the third figure from the top. There is severe aggregation of the protein. On the other hand, the presence of asulfame in the glycation system prevented this was in terms of measured in terms of size. We could show that the size of the aggregated particles was around 2000 to 5000 nanometer. On the other hand, the size of or the less aggregated or prevented aggregates was 50 to 300 nanometer or 50 to 200 nanometer. Then we did docking studies to realize that to analyze whether there is a similar binding pattern on the BSA for the BSA glucose and asulfame. We could show that glucose binds with BSA, binds to BSA around the similar kind of amino acid residues like serine 192, histidine 145, proline 146, arginine 144, and aspartate 108. If you can see that we have almost the same amino acids which are also the binding site for the asulfame potassium. So coming to the conclusion, we could show here is that glycation induces the alteration in the structure of the proteins and DNA, and there is a formation of early and advanced glycation products that are quite harmful to the body. Then the presence of thymoquinone and asulfame potassium could potentially reduce early and late glycation products, total ages, and the protein aggregation. There was also a reversal of the glycosylated damage of DNA. Fluorescence potassium conclude from this study that to be a glycating agent can be used to prevent the glycation-induced processes like aggregation and glycosylative damage. We need to further characterize the anti-glycation property of these compounds and the functional groups which are involved and the products which are generated. So this work has been done by my students, PhD students Dinesh Kumar and Prerna. I have received funding from, I acknowledge this receiving funding from different government and non-government agencies. I have my collaborators, and thank you for being a patient listener. So if there is any question, I would be very happy to.
Dear Ahmet Ali, thank you very much for your valuable presentation. It was very nice to do something together here in this platform. And is there any question? Is there any question to the presentation of Dr. Ahmet Ali? Dear Ahmet, if it is possible, Dr. Stefan has got a question. We are listening to you. Your voice is open.
Yes, yes. One question for Mr. Professor. So if it is too much glucose in the blood, like people eating too much sweets, this too much glucose in the blood may block also the antibodies and thus reduce the immune system power?
Yes, the presence of glucose also affects through this glycation, which I am talking about, antibodies also get glycated. So that could be one mechanism for immune suppression. But there is also another mechanism that is through the generation of ROS and RNS that goes through anti-inflammatory agents. So immune system is compromised during the glycation process also.
Okay. So no more Coca-Cola.
Yeah, too much sweet is not good actually, but glucose is a very wonderful molecule. I always.
Yes. Honey is okay. Honey is okay.
Yes, thank you. Yes, very true. Honey has lots of potential without having much of the adverse effect.
Yes, yes.
Thank you very much for your valuable presentation. And dear respected participants, we are going to make a break for the lunch. We will continue 14:30 again here. The next presentation is going to be moderated by Professor Dr. Otile Bovis from Cluj University, Romania, and we will be continue with her moderation in 40-30. And during meanwhile, we have prepared some music and poster presentations for you. Enjoy the music and see you soon after the lunch.
Thank you, doctor. Thank you. Yes.
Thank you.